Good morning, and welcome back for round two. Thanks to everybody for taking your Saturday morning to join us for what I think will be another very interesting and inspiring discussion of some issues surrounding pediatric research ethics. Just a couple of housekeeping announcements before we start this morning. Kathy Cohorn wanted me to mention that you all have a CME certificate in the back of your books, and you need to combine that with making sure you sign in each of the 2 days. So if you signed in yesterday, make sure you sign in again today at some point before you leave so that you get credit for CME activity. The other thing I just want to mention, particularly for those of you who are from out of town and maybe trying to catch planes, traffic is going to be interesting in Seattle this weekend. It's interesting all the time, but the westbound, all but one of the westbound lanes on I-90 is closed, and Highway 2 is closed because of fires on the other side of the pass, which means that there will be a lot of traffic. Anybody headed west over the passes is going to be on I-90, and they're going to have trouble getting there. Or getting back. And so that's going to make for some interesting traffic flow. And when you combine that with the Sounders game, that's our soccer team, that's starting at 1 o'clock on the south side of downtown, and Bite of Seattle, which is occurring at the Seattle Center, and something called Dota 2 Championships, which is also occurring at the Seattle Center, and I understand is bringing in thousands of people. Traffic's going to be a little interesting downtown, so you might want to just sort of plan ahead a little bit if you're headed to the airport. Take the train, exactly. That's the one way to avoid all of it. All right, so we have a great morning scheduled. The way our morning's going to work is we're going to have a couple of talks, and then we will have breakout sessions, and I'll tell you more about the breakout sessions just before the break. So that you can make your final choices. And then we'll come back here for sort of a final wrap-up talk by Alex Capron. As I said, I think it's going to be a really stimulating morning. But starting us off is one of our own, Holly Tabor, is an associate professor of pediatrics in the Division of Bioethics and a colleague at the Truman Katz Center for Pediatric Bioethics. Holly came to us from Stanford University, where she did her PhD in epidemiology with a minor in genetics, and then did her postdoc with the Stanford Center for Biomedical Ethics. And Holly has been really thoughtful investigator in looking at issues related to return of results, and many of the issues that, since our last conference 10 years ago, are relatively new in terms of sort of the kinds of issues that genetics and genetic technology is starting to present to IRBs and investigators and those doing research. So Holly's going to talk to us this morning about Beyond All or None, Return of Secondary Results from Research and Clinical Exome and Whole Genome Sequencing. So Holly.
Thank you, Doug, and thank you very much to all of the organizing committee for the invitation to speak with you all today. I have incredibly fond memories of the conference from the years that I've been able to come, and so it's a real pleasure to be able to talk about some of my work with you today in the context of the broader theme. I will say that it's hard to be the first talk this morning after a wonderful day, and particularly very moving session at the end yesterday, but this will be something quite different for us to focus on. So I'm going to talk today, starting out, I know for some of you may already know this, but with a brief review of what are exome and whole genome sequencing and why are there important ethical issues about returning and offering results from whole genome and exome sequencing. Then I'm going to talk a little bit about what we know about what results people might have from exome and whole genome sequencing, because that really does affect how we think about the ethical issues about what we might offer in return to anybody who has their exome and genome sequenced, especially children. Then I'm going to actually dive into what the ethical challenges are, specifically in the context of pediatric research, because that's what we're mostly here to talk about this weekend. And then give you some data from one of the studies that I've been involved in about what parents think about different sequencing results from exome and whole genome sequencing for their children and what they would want. And these are parents who actually are having exomes for their children done as part of research studies. And at the end, I'll have a few seconds to just briefly kind of tease you and talk about what I think some of the emerging and future challenges are related to results from exome and whole genome sequencing, especially in research, but also in clinical care. So this slide is partly an excuse for me to show you my pretty picture. And I think most of you in the audience probably already know something about genetics, and that's also possibly something that was different from 10 years ago when the conference started. But as you all probably know, DNA is made out of 4 molecules called bases, and they have symbols A, T, C, and G. And they're linked together to make a standard DNA that forms that famous structure, the double helix, which kind of looks like a twisted ladder. And one of the things that really my work focuses on is thinking about genetic variation, and there's genetic variation among all of us, and essentially genetic variation is largely created by copying errors when DNA is copied in the process of meiosis, and this is an example of the sequence from 5 individuals, and you can see actually the variant is pointing to the wrong variant, but you can see the green T's and the C's. That is not a variant. The green T's and the C's are a difference where 3 of the individuals are homozygous for a T, the 4th individual is probably heterozygous for a C and a T, and the 5th individual is homozygous for a C. And so that's the kind of variation that we're looking for. Some of that variation is just natural differences between all of us that don't have any changes on phenotype at all, and some of that variation causes different phenotypes, diseases, risks for diseases, and so forth. So over the time that I've even been working in this field, there have been huge technological advances that have made a substantial impact on how we find genetic variation. That picture on the left in 1987 is gel sequencing and Sanger-based sequencing, which was the way they were doing it when I started out in this field, and you actually had to do this very labor-intensive process of pouring gels and manually loading in each sequence. I don't know if anyone here has ever seen that or done that. It was very, very time-intensive. It was what they were doing at the start of the Human Genome Project, and they had people working 24/7 in centers across the country to try to actually get the volume they needed to complete the Genome Project. And it was sort of on the order of hundreds of base pairs a day. Jump ahead to 2012 or now, and now we have what's called next generation sequencing. Very, very different technology, very non-manual, very, very automated, and essentially much, much larger numbers of base pairs that we can get through a day, and we can do exomes and genomes. The actual sequencing part takes a matter of days. The prep part included takes a matter of weeks. So it's just a very, very different order of magnitude in terms of the amount of data that we can generate. And this is a graph, and the y-axis is the number of kilobases per day per machine, and the x-axis is the years, and you can just see how exponential the growth has been in the amount of data that can be generated and the amount of exomes and genomes from individuals that can be examined at any one time, either for research purposes or for clinical purposes. So it's really, really, really changed the way we think about the research questions, and it's really, really changed the way we think about the ethical challenges. And that's been really interesting and really where a lot of my passion and interest lies. So exome and whole genome sequencing use these next generation approaches to sequencing. And exome is the parts of the DNA that make up the proteins, and that's only about 1% to 2% of your genome. And I could give a whole nother talk about what the other 98% is, but I won't. And most but not all disease-causing variants and even risk increasing or decreasing variants are in the exome. So really, that's what people have focused on. It's a little bit easier in some ways, it's a little bit higher quality in some ways, and it's a little bit cheaper. In the 6 years that I've been here at Truman Katz, exome sequencing has been the focus, although that's starting to change and moving more towards whole genome sequencing, which is actually exactly what it sounds like. It's looking at the whole genome. And it allows researchers or clinicians or labs to look at variants not just in the coding regions for the exome, but also the non-coding regions where there are some really important changes and you can get a bigger picture. So these are both things that are happening right now in research. Exome sequencing is whole genome sequencing is really not happening in clinical care yet unless it's in the context of a research study, although that is and will be changing any day now, I would say. By the time this conference happens next year, I would suspect that whole genome sequencing will be available for clinical service in some settings. So the way I got involved in this is when I started here at Truman Katz, I had a project from NHGRI where I was being what I called the integrated ethicist, working with the Center for Clinical Genomics and Genome Sciences at University of Washington. And as a part of that, they actually did a study where they applied both exome sequencing and whole genome sequencing for the first time to try to solve a Mendelian condition. And it was really the first family study that used these new techniques, and they intentionally chose to study a rare limb malformation disorder called Miller syndrome. And this is the family that was studied, and they have given permission for this picture to be used. And what was so exciting about this project was that they actually were able to identify the mutations in the gene that actually caused Miller syndrome. And so it was an example of the potential success and power of this approach to solve rare unsolved Mendelian disorders. But this family actually had another sort of subphenotype where they actually had a lung condition that presented a lot like cystic fibrosis. And in fact, at one time, some of their clinicians thought it might be cystic fibrosis, but they couldn't identify two causal mutations in the family. And they had been quite ill with it, and they had been hospitalized quite a great deal for it. And in doing exome sequencing, without sort of intentionally trying to, and it was, sorry, backing up, it was unclear whether or not that lung phenotype was something related to their Miller syndrome or was in fact something else that was going on with them. And in the context of doing the exome sequencing, actually as part of the methodology of looking for the gene that caused Miller syndrome, they actually identified that this family had another mutation in a gene called DNAH5 that causes another rare recessive condition called primary ciliary dyskinesia, which is a lung phenotype where the cilia don't work properly, and the pulmonologist in the audience can correct me, but it presents frequently with a lot of similarities to cystic fibrosis. And so this was really important to the family because it was really a condition that was affecting them and affecting their lives. And it was an example of sort of a real incidental finding, an example of something we weren't looking for, but was something that was found in the course of the research that absolutely had clinical importance to this family. So from a sort of ethics standpoint, this was really interesting because it was really part of what we're going to have to struggle with on a larger scale as we do more exome and whole genome sequencing across settings, as I'll talk about in a second. So as part of my role in that study, I actually designed and conducted a parallel empirical study where we actually interviewed the two families that were involved in this study at different times in the process after informed consent and after they received results back. And I'm not going to present all of the data from that, but just briefly, the family was really challenged in thinking about what kinds of results they did and did not want to know when they could think about the range of all possible results. And this was before they got their results back. They really didn't believe that results would substantially impact their daily lives. They hoped that the result, cause of Miller syndrome would be found, but they still really wanted the option to receive all results across a range of categories and a range of possible conditions and risk factors. And they expressed that they really wanted control and options for choosing different kinds of results, for changing preferences over time about what results they wanted, for viewing results at their convenience and being able to ask questions over time as they process the information. And they wanted to be able to share the results with their healthcare provider. And again, they really wanted to be offered a range of results. And so these findings, while they were only on a very small number of families in a fairly new and novel study, really helped frame sort of a research agenda for what we've been working on for about 5 years since then, thinking about how can we facilitate allowing individuals to think about the range of results that might be identified and what they might and might not want, and maximize the benefit and minimize the harm from those kinds of results. And I think one of the most important questions that has come up, which I think is interesting because it's an intersection of sort of understanding the science and understanding the ethical issues involved, is thinking about, well, what results are people really going to have? What are we going to find? And I think, in the past, before research studies were done with sequencing, people didn't think we were going to identify very many results in the context of linkage studies and other kinds of genetic studies using other approaches. And so the answer from the large exome studies that have been done in the last couple of years is that it depends, which is, a really frustrating answer, but it depends on a couple of things. It depends on how you define or classify results. Do you define or classify results in terms of how useful they are, or what their utility is? And even then, how do we define utility, and how do we define what's useful? Do we define them in terms of what's actionable? And a number of our colleagues actually think that that should be the focus, something that I can do something about to mitigate or prevent disease in a short time frame. Or do you define them in a different way? Can I diagnose something that I might not know about otherwise? Can I predict an outcome? Can I modify my risk over a long period of time? Do I only return results that are highly penetrant? How do you think about those things, and how do you think about explaining those to individuals who have had exome and genome sequencing? It also depends what genes you look at, as I'll show you in a second. It depends who you look in. It depends a lot who you look in. The results that we might find among this half of the room might be different than the results that we would find in this half of the room, especially depending on the demographic characteristics. It depends what criteria you use, and this is a great debate, as Steve and Lainey know well in our field, about how we're actually going to define criteria that we can sort of standardize across institutions and labs, and how many reviewers you have. There actually are some studies that show that it depends on how many different people you have studying it, which is true for a lot of things in medicine. And then I'm particularly interested in how we think about utility in kids. Do we think about just short-term medical utility, something that's going to affect a diagnosis or a condition that they might have during childhood very soon? Do we look more at long-term medical implications, including some conditions that they might develop in childhood, but they might not develop until early adulthood or later? Or maybe only conditions that they might only develop in adulthood. What are the risks and benefits of that information? As I'll talk about, we've talked about that one way in the past, and we're sort of starting to think about whether we need to think about that differently. Reproductive planning. Do you give kids information about carrier status when they're not likely to make the decisions using that information for some time? And broader life planning. So I'll give you some examples of those from our research in a moment. So just to give you a quick view into some of the data, there have been a number of studies looking in large populations about what results people have. So if you focus on a more narrow definition of utility, highly penetrant genes and variants that actually result in rather severe phenotypes, there is a study that was just submitted looking at what's called the Exome Sequencing Project 6500, which is 6,500 exomes from a range of studies from the National Heart, Lung, and Blood Institute, which is not that relevant to this discussion. And if you just focus on what the American College of Medical Genetics, I'll talk about this in a second, defined as their sort of most actionable genes, which was 56 genes, they found that 1.5% of European Americans and 1% of African Americans had a result that would be potentially offered for return in those categories. So that's not very much, although for those people, those results might be very important. When they expanded their criteria a little bit to 116 genes that they defined as medically actionable genetic disorders that may be undiagnosed in adults, and this has been submitted and was presented at ACMG earlier this year, it went up a little bit, 1.9% in European Americans and 1.2% in African Americans, but not that much. And then a study by Les Biesecker and his colleagues at NIH, which is called ClinSeq, looked in just under 600 people, and they looked just at cancer risk genes, 37 of those, because people might be very interested in those, and about 0.8% of non-Ashkenazi Jewish participants and 4.1% of Ashkenazi Jewish participants actually had returnable results, actionable results in those cancer risk genes. So again, you can kind of see that it depends on your definitions, how you look, and who you look in. But if you broaden your definition of utility to think not just about those highly penetrant, highly actionable things, we actually have conducted a study that was just accepted in the American Journal of Human Genetics, looking at 3 different categories of genes that are a little bit different than what we've usually thought about, but using very, very conservative criteria for how we define what the genes are and what the variants are that we are fairly sure there's substantial evidence that supports them actually affecting risk and affecting pathogenicity. So if you looked at genes for newborn screening conditions, and the good thing about newborn screening is that we really understand the biology very well, and we have very good databases about the variants and whether or not they're related to disease, and we also have very good population estimates in this country. So we looked at 39 newborn screening genes and looked at known pathogenic variants for those conditions. And when you look at those, just at carrier status, these people don't have the conditions. 41% of European Americans and 44% of African Americans carried at least one known pathogenic variant in those 39 newborn screening genes. So that's a very different, that's sort of the other end of the spectrum. When you look at a condition that's late onset and is a complex condition where there are lots of genetic and environmental factors involved, but again, about which we actually know a fair amount about the biology and the genetics involved, age-related macular degeneration, we looked at 17 genes for which there is clinical genetic testing available, and we looked again at things for which there was substantial evidence in the literature for known risk variants, and when you look at those, everybody's got them. 100% of European Americans have at least one, and 99.8% of African Americans have at least one. So again, different end of the spectrum, perhaps not that useful, you might argue, and we can talk about that, but different end of the spectrum. And when you looked at genes associated with medication response or pharmacogenomic genes, and we use a database called PharmGKB, which actually curates data on pharmacogenomic risk, looks at lab evidence, population evidence, and we use the highest level of criteria of causation. Again, almost everybody has at least one of those known variants. Now, almost everybody may not ever come in contact with those drugs. Some of them are chemotherapeutic agents, some of them are blood thinning agents, other kinds of things. But for the people who will come in contact with those drugs or may, that information is potentially very useful and might avoid adverse responses or not the response that the physician was intending. So everybody's going to have results of some kind. It depends on how you define it, which is, sort of a duh. Of course we all have results. But they're results about which we're not guessing. They're results about which we actually know something. And so it's really no longer a question of whether results of clinical or personal utility will be found. As a result, I argue that we need to shift towards thinking about how to interpret and manage different kinds of results and really manage results expectations and obligations of research participants, researchers, clinicians, and patients. And any analyses that we do of results and of genomes and exomes will be dynamic. Our existing databases, some of them are good and some of them are not, are very dynamic, and our knowledge is limited and we're learning more all the time. And our interpretation can be highly subjective and context dependent. And really our understanding of disease etiology and pathogenicity is really changing. And we're finding that we think we know that these changes in this gene cause this sudden cardiac death phenotype, and then we look in these big populations and it turns out that a much larger proportion of individuals may be walking around with these than we thought had long QT syndrome. So we really have to be careful and think about how we interpret these and how we understand them. So thinking about pediatric research, we have to think again about what are the obligations to offer a child's parents different kinds of results. So last year we talked about RET mutations for medullary thyroid cancer, where there's actually a very clear action to be taken. People often know they have a family risk of this, but sometimes they don't. Is that the kind of result that you would want to offer to a parent? It is on the ACMG list of genes. How about a BRCA1 mutation? Probably not going to be relevant until adulthood, and there's been some work by Ken Tersiak and others about whether or not adolescents should be tested for this. A KCNQ1 mutation for long QT syndrome, again, not likely to present until adulthood, although there are, I believe, some presentations in the late teen years. SCN1A mutation for epilepsy could be very, very useful for both diagnosis and for decisions about what treatment would be appropriate for a child presenting with epilepsy, and is actually done quite frequently as a part of standard clinical workup. Or a CFTR mutation that may not actually cause CFTR, but may cause some of the other phenotypes that are associated with mutations in the cystic fibrosis gene. And so I think one of the big questions is, does the duty to warn a child or a family potentially outweigh what we call the sort of right not to know, or the right of a child to make the decisions for themselves when they're adult about whether they want that information? And is this question, or is the duty to warn, the same or different for different kinds of results in different people? I think I'm as guilty of this as anybody, tend to sort of think about these things as equally applying to all results and all people in all contexts. And as I'll show you, I don't think that that's necessarily true, which unfortunately makes making policy much more complicated. Does the child have a right to access that information as an adult? At the end of the talk, I'll just give you an example of whether or not children actually should be allowed, even if we decide not to offer certain information to them as a child, if that information is being used by researchers, or Pearl will talk about this later, in a biobank later, do they have a right to some of that information once they're old enough to make that decision? And how do we balance autonomy, beneficence, and non-maleficence? So just briefly, we are very lucky that Lainey's here. I just want to mention that the AAP ACMG guidelines that were written together for pediatric predictive genetic testing came out last year, and this was an update of sort of existing guidelines that had been placed but not updated for some time about genetic testing in children. And I just wanted to put these two quotes here, that decisions about whether to offer genetic testing and screening should be driven by the best interest of the child, and I think that's something that probably everybody at this conference would agree with. And then went on to say the predictive genetic testing for adult onset conditions should generally be deferred unless an intervention initiated in childhood may reduce morbidity and mortality. And I think that has sort of been something that people have been very comfortable with in the sort of clinical genetics field in general, that we wouldn't necessarily order a genetic test that was only predictive for an adult onset disorder in a child. But again, the challenge comes up when we're not ordering that test, but that information becomes available in the context of clinical sequencing that we've done on a child for a condition that actually is occurring in childhood. And around the same time, as I mentioned earlier, the American College of Medical Genetics came up with their guidelines for incidental findings reporting from clinical sequencing, which has been very controversial and much discussed, at least in our field. And essentially, just to summarize, they listed 56 conditions with what they called mandatory reporting, although they've revised this a little bit now, which were essentially those most likely to be verifiable by other diagnostic methods and amenable to medical intervention. So they were really focusing on that sort of actionable definition and a sort of fairly strict definition of what actionable was. And they sort of, some argued, came up with a slightly contradictory conclusion to the AAPA-CMG conclusion, which said at this moment in the evolution of clinical sequencing, an incidental finding relevant to adult disease that is discovered and reported through the clinical sequencing of a child may be the only way in which that variant will come to light for the parent. So they were actually arguing that adult onset results should be returned in children because it had benefit to the parent, which I actually disagree with that as the primary reason for returning the result, and there's been a lot of discussion and debate about that. I'm more interested in whether or not offering or returning that result has any short-term or long-term benefit for the child, and it's usually the parent who's the broker of that decision. So, and then in April of this year, they released a sort of frustratingly vague statement saying that individuals would be allowed to opt out of secondary results, and they didn't explain how or when that would happen, and they didn't provide any information. And I have heard, and others may be able to clarify this, that they are working on it, so there will probably be more detailed guidelines soon, but they're no longer mandating return of results for any secondary results from clinical sequencing. And the labs are not completely following these guidelines. Some of them are and some of them aren't. So this has been, again, a topic of much debate in the field, and as a part of this, our group did a survey, online survey of genetics professionals, not just clinical but also research, members of the American College of Medical Genetics, the American Society of Human Genetics, and the National Society of Genetic Counselors, and we got just under 800 to respond, and we asked them what they thought. We asked them a lot of questions, which I'm not going into, but I just wanted to show you these. We asked them what they thought about this issue, about what we should do about results in children, and we also asked them questions about how that related to their practice. And 88% agreed that incidental findings about childhood onset conditions should be offered to the parents of a child who's undergone medical exome sequencing. But somewhat surprising to us, 62%, which was more than we were expecting, thought that incidental findings about adult onset conditions should be offered, not necessarily mandatorily returned. And in fact, they didn't agree with mandatory return, but offered to the parent of a child who undergoes sequencing. And they also felt that 62% thought that carrier status results should be offered. But again, 20% did not think they should be offered. So this was interesting to us to sort of get a finger on the pulse of what a lot of the people in the field are thinking, and we have other data breaking it down by discipline and specialty within the field. And that paper came out 2 weeks ago. I hope I've sort of shown you that this issue about what results we're going to find, what results return, kind of creates this elephant in the room. I think we now have to sort of think about and talk about what we're going to do about these results and the ethical framework about how we're going to think about the potential benefits and risks of the results, both in clinical sequencing and in research sequencing. So I actually paid for a license for this cartoon because I like it so much, and I think about it every day now. And the guy was very nice about it when I told him I was going to show at this conference. So we are doing a study that's a R01 study funded by NHGRI as part of the larger Return of Results Consortium sequencing sort of program that they have, which is called the My46 study. And I'm not going to go into My46 today. You can look at it online at my46.org, but it's essentially a web-based tool for allowing individuals to set preferences about what kinds of secondary results they want to receive, and actually receiving those results online with follow-up from a genetic counselor compared to sort of standard of care, which is getting them from a genetic counselor. But I'm not going to talk about that part of it today. I'm going to talk about what parents said about results. And we recruited about 150 participants who were already enrolled in existing studies of Mendelian and complex diseases and whose exomes had already been sequenced as a part of that research. And as I said, they were allowed to choose from a range of different results that they wanted, and then they're being randomized to receive results through these two different methods. And this is just to flash up who these people were. About 60% were parents of minors who we talked to, a range of different phenotypes. I think those phenotypes are important to some of the results I'm going to show you. We mostly had women who were interviewed and responding, and the average age was 45 years, and they were predominantly self-reporting as white and higher SES. So we asked the parents about what they thought were the benefits of secondary results for their affected child. And several of them said that they thought that it would help them prevent future conditions. So one cystic fibrosis parent said, knowing things that could be enlightening towards preventing or correcting lifestyles, anything that would make her life easier. Another parent of a child with a brain malformation disorder thought that secondary results would help guide or ease management, and said, I want to know everything that's going on so I can know how to treat it or what the doctors to see or know how to relate to him. A couple said they really wanted the results for their or their other children's future reproductive planning. A parent of a child with limb malformation said, I'm curious if what she has, mostly if she would be able to pass it on to her children. That's a big one. And quite a number of parents brought up reproductive planning as both related to the condition and related to secondary results as something that they would explicitly want. Many of the children, as you probably saw from the brief slide on the phenotypes we studied, have some form of disability. And a number of parents talked about secondary results relating to the planning for the long-term future for their disabled children. This one parent said, well, obviously there's going to be a time in our lives when we're not around and our son is still alive. So if we have some insight to what his future medical needs might be or some things to watch out for, we can advise survivors to keep an eye on this, including healthcare providers, and family members. So really focusing on what to tell people to look out for. They use interesting metaphors when they're talking about this. And this one other brain malformation parent talked about sort of tea leaves and crystal balls and said this somewhat tongue-in-cheek in the context of the actual interview, but was serious about it. Our son is nonverbal, so we have the added trick of he can't tell us how he's feeling. So if he would develop symptoms, we would have no idea. He's a bit of a puzzle. We're reading tea leaves right now. So if there's diagnostic information we can get to tell us stuff that he can't tell us, that's to our benefit. Because it's for a dependent child, and I would really have to think about, estate planning and his future needs when we're not here. I think we would really want to know everything. We could use the help of a crystal ball, I guess. So they look at the future, and they feel like they don't know what's in store for their child in any way, both related and unrelated to their condition, and they want information that will help guide planning, care, prevention, and treatment, not just for them, but for the people who are taking care of their child who at least to some degree lacks some capacity. A number of parents talked about the potential benefit of both primary and secondary results to the siblings of an affected child. Several talked about information for siblings' health, and this one parent said, as far as offering a heads up, since he's a direct sibling, he could have the same thing, so I guess that would be a benefit. There's something we need to look at for him. And that was also motivating them to participate in research. And again, as I mentioned, they talked about informing siblings' future reproductive choices. And this parent of a child with autism said, I think we're interested in the information that we can pass on to our daughter and our son, affected son, if they're potentially going to have a family. So they're thinking about the long-term implications as well as the short-term implications. And in general, their attitude was that they really, as I'll show you in a second, they really felt like whatever the results were, they could handle it, they could cope with it, and they really would rather receive results than saw information as good. Again, in this particular population of people that we talked to. And this mom said, I guess it makes me a little nervous, everything we've gone through. I just don't want her to have anything else. But I also feel like if there was something that we could prevent or make better, I would want to do that. So I guess I feel pretty relaxed. Whatever, we've made it this far. We've made it through all those things that have gone wrong. So, it's like bring it on, we can handle it. And there were several parents who sort of expressed that same sentiment. A couple of parents talked about the child's role in decision making and talked about how they sat down as a family to talk about what they would and wouldn't want. And one mother of a child with autism actually talked about partnering with her child and said, I think there's a possibility for me partnering with my son in his own care with information about himself, since science is interesting to him. So that was very interesting and important to us. One cystic fibrosis parent said that her child's view had been very different from what her own view had said, and basically said, I want to know things about me so I would know then when I grew up and got married and had kids, that's kind of where her mindset is, about her future, and I hadn't even thought about it. So having that conversation with her child actually influenced what she chose to receive on behalf of her child. And then another parent talked about preparing her child for adulthood and taking responsibility for their own health, and said, the opportunity to try and prevent or reduce the risk later in my son's life, or educate him, since I can only tell him so much, he's going to have to use that information, make sure he always goes to his doctor for his checkups. And that's consistent with some results from the literature about returning other kinds of genetic results that are risk kinds of results. So this is the preference grid that they saw, just to show you that you can't really see this, but they had a range of different disease categories and carrier status, medication response, and ancestry that they could choose what results they wanted to receive. And just briefly, I want to tell you, 80% selected all possible secondary results, and a slightly larger proportion of parents than adults selecting results for themselves chose them all. The average number of result categories they refused was 1.8 in parents and 3.5 in adults, so adults were refusing more kinds of results for themselves. And in parents, the most commonly refused secondary category was ancestry, whereas in adults it was brain and nervous system, and so we're analyzing more of the qualitative data about that right now. And interestingly, thinking about parents refusing results for their child, one said that their child should decide about carrier status when he reaches adulthood, and said, I didn't really feel like it was any of my business. I thought it was more his thing. So that's kind of consistent with thinking about how we think about deferring that to adulthood. And then this other parent with brain malformation said no to cancer as a category, and said, I just wanted to let him be a kid and not have to worry about that. So those are the kinds of reasons that parents think about why they would not want to receive results, though again, that was the minority of people in our study and from the populations that we studied. So in summary, parents really want results both in our study and the kinds of phenotypes we studied, which is an important limitation, really wanted results of short and long-term clinical utility for management of their child's health, prevention and mitigation of future health risks, and promoting healthy behaviors and responsibility. They really see getting secondary results back as part of being good parents and as part of long-term life planning, and they use good parent in some of their quotes. And they really want to promote risk awareness for their children's current and future caretakers, and they talk about estate and long-term planning for children who have significant disabilities. They talk about health benefits for siblings, and that being sort of a driver for some kinds of results. So they see the benefit outside of the individual child, which we can talk about the potential pros and cons of whether that should affect decisions about returning results. And they are explicitly interested in results for reproductive planning, even though obviously that doesn't have a short-term clinical benefit or isn't short-term actionability, both for the affected and the unaffected siblings. They really want to involve their child in decisions as appropriate. They want results, but they want the ability to choose and defer some results until later. They may view results through an optimistic lens. I think that's an important finding here, that they see, in other quotes, they sound more realistic about it, but they are very hopeful that research will lead to positive things for their child, which I think is an important follow-on to what we were talking about yesterday, about how we talk about integration of research in clinical care. They really acknowledge, almost to a person, that they might get worried or anxious about the results, but they really do also say at the same time that they think they can work through it. And they think the benefits outweigh the potential risks and harms. And I think it's important that the perspectives may differ across pediatric populations, and this is one subset of populations. We need to talk to other kinds of both disease populations and healthy populations to look at the perspectives and think about the benefits and risks across populations. So finally, I just want to set an example of the emerging challenges of what happens with exome and whole genome sequencing in pediatric research. There was a press release just about a month ago that Autism Speaks and Google have decided to put 10,000 autism genomes into the Google Cloud and make it publicly available for research to try to promote and stimulate research into autism. They're planning to put up both genomes and phenotypic data, and it's funded by Autism Speaks, which is one of the national advocacy groups. But they are not currently offering primary and secondary results to individuals who are enrolled in these studies. So they're it's interesting, they're putting the information on the cloud, making it available, but they don't have the resources or the structure to actually offer any kinds of results to the families who are enrolled in these studies. And when this came out, the Autism Speaks co-founder, Bob Wright, was quoted as saying this announcement represents an unprecedented intersection of business, science, and philanthropy that will drastically accelerate the pace of autism research. Very optimistic. But at the same time, in the autism rights community and autistic self-advocacy self-advocate community. I mean, Amy Sequenzia, who's a fairly well-known autistic self-advocate, said, this should not be an excuse to use our genetic information for the profit of others. I notice there are no plans whatsoever to use profits for better services. So there are issues about consent, there are issues about use, there are issues about what the primary goal is. I think this also relates again to our discussion yesterday about what happens with data from research, even if it is being used first for clinical benefit potentially or for learning, but also examples about return of results. You know, if we're making information available to everyone, what obligations in research do we have to make at least information we understand well available to participants and to pediatric participants? So I think most pediatric research will include exome and whole genome sequencing, and we have to think about these obligations and think about how we manage results and think about whether children should have the ability to access results when they reach adulthood, especially if they're shared in places like the Google Cloud. Does it matter if the children are sick or healthy at the time of the research? I think maybe it does. And does it matter if primary results are being offered for the condition that's being studied? And I think that also raises some really important questions. And do we need to think about or rethink the benefit and risk ratio of results? I think we need to think about, can we do harm in some situations by not offering some results? And how can we better explain the benefits and risks of specific results at different ages and time points, because they may not be the same across all ages and all populations. And then I think we absolutely have to assess and track the benefits and harms over time. I and others can stand up here and present our small studies and hypothesize and speculate, but I think we really need data in order to do a good job in thinking about how we actually should implement this in the best way possible. And I work with a lot of wonderful people. This research is very interdisciplinary, and I couldn't do it without many people, including a lot of the research staff here at Trim and Katz. And the data I presented from the exome sequencing project is an even larger number of people, and without all of those people, none of this would be possible. Thank you.
Thanks, Holly, for a great start to the morning. We will give you all opportunity to ask questions and comment on both Holly's talk and our next speakers following these two. And our next speaker you met yesterday, Pearl O'Rourke, as I mentioned, started out as an intensive care physician, was the director of the ICU at Seattle Children's for a while, and then changed her career path about 20 years ago now, I think, and went into health policy, and has an incredible range of experience, both in terms of the regulatory community as well as clinical practice that she brings to her work in research ethics. Her current position is as Director of Human Research Affairs at Partners Healthcare Systems in Boston, and she also is an Associate Professor of Pediatrics at Harvard Medical School. Pearl?
Good morning, and it's wonderful to be back in Seattle. I was very happy until Ken has now made me nervous. It's going to take 5 hours to get to the airport. So I'm going to talk very, very fast so I can get the hell out of here. Okay, so biobanking is on everybody's mind, and I hate to think how many hours a week I spend on this. And for the agenda here, you can read as well as I'm going to spend very little time on why this is important and spend more time on the heartache. And the last bullet is most important. Holly has already covered, I think, what is one of the most challenging of all of these, so thank you very much, Holly. So perks and promises. What does a biobank do? Research needs tissue upon which to do research. And particularly with genetics, the need for tissue has just expanded. So we're going to find out what stuff runs in families. We're going to bring every disease down to a protein level. We're going to understand why it happens in various people and others, with the ultimate goal of better health through better therapeutics and diagnostics. Another thing that always comes in when you talk about tissue banking is personalized medicine. So I just have a few slides here. This is my approach to personalized medicine. Since I have the microphone, this is your approach to personalized medicine. These people like personalized medicine, therefore so do we. That's Francis Collins on your far right, who is the head of NIH, and since he signs the checks, sort of, for research, and he likes personalized medicine, we like it also. So there's a lot of national leadership push for the benefits of this. The best definition, Wikipedia. This is my number one resource for everything. Well, it's so easy, you know. If you can spell, you can get there. But I do think this is a good one. Use of detailed information about a patient's genotype or level of gene expression, their clinical data, in order to select a medication, therapy, or preventative measure that is particularly suited to that person at that time. Who would not sign up for that, right? So how does it work? You have a disease of interest, the green blanket disease. You collect information from every patient who comes in with this disease. You get tissue. You do really detailed demographics, comorbidities. You follow them in perpetuity. You look at their families. And you look at how do they respond to various therapies. How does their disease progress? And what you find out is subpopulations emerge. It might be that, you know, those who live in the Seattle area are the only ones who are really affected. The rest of the country doesn't even have this disease. How they differ in response to therapies and comorbidities, et cetera. And then what you can do is study these subpopulations. And you look and see, is there a genetic marker for these subpopulations? If there is, you may actually be able to develop what they call a theranostic, which is that last word on that last bullet there, which would be a test to say, okay, you come in with green blanket disease, we're going to do a genetic test on you, and lo and behold, you're one of the light green blanket disease people, and for you, this is the more correct therapy. This is already being done a fair amount in cancer research. And some of the difficulties of cancer research is that you're looking at the cancer genome, not the genome you're born with. And what I found is that with therapy, that cancer genome changes. If you also look at a primary tumor and metastases, you may have different footprints. So it's not, you know, a black and white situation. For multifactorial diseases such as hypertension, you're going to need hundreds of thousands of people. Hence, this is why we're here talking about tissue banking. That's it for the positive. Now onto the more fun. So the problems. Access to tissue. What we do versus what people think, or some people, quote, know that we do. How many in this room have been a research participant? Pretty good, okay, but not, how many have not? Okay, I'm going to ask this again later, and I'm hoping that everyone's hand goes up, because you think you haven't been a research participant. How tissue gets into research is driven by the common rule, which is a federal law by which we define what research is, what human subjects are, and if you're federally funded, you got to follow these laws. And the definition of human subject is a living individual. HIPAA, which we also have to follow, also covers dead people. Okay, so you already see the regulations are always in conflict. But for the common rule, you have to be living, about whom an investigator who's doing research gets data through intervention or interaction. It could be a biopsy, it could be a survey, it could be a telephone call, but you somehow are interacting, or identifiable information. Your medical record is a human subject. Now, like everything, you got to take those laws and put it into more practical approaches. So you look at these two basic questions for tissue research. Why was the tissue obtained? And regardless of how it was obtained, is it identifiable? So to walk you through this, I hope, there is no pointer. Okay, that's okay. So the first question you ask is, why was the tissue initially obtained? If it was obtained solely for research purposes, there has to be an IRB action and informed consent. You can't sneak up on somebody and get a tube of blood and say, oops, I'm sorry, was that your blood? You know, so there's an interaction, and of course you have to get consent. Why is anyone going to give you a piece of themselves without you having that interaction? But let's look on the clinical care side. We have Ken here, who I sat next to him at dinner last night. I saw what he ate, and you know, if I were his physician, I'd haul him in for a little cholesterol and lipid profile. So in he comes, and what happens is we're going to draw 10 cc's of blood. They only need 2 cc's to do the test. Why do they draw 10? Well, the tube's that big, you know, there might be some spillage. The first result might come back and go, whoa, no living human could have this. We need to repeat it. So they don't have to schlep them back in. They can repeat it. So it's not for nefarious reasons. It is solely kind of just logistically makes most sense. But after they do the little test on the 2 cc's, there's 8 cc's left. For those of you poor at math, 10 minus 2 is 8. Okay, so you have 8 cc's of blood left. What do we do with that? We usually trash it. Every institution has some amount of time. Ours, it's 6 days, just to make sure there's no other things that have gone wrong, and then the stuff is trashed. But what could happen is we could use that for research. So following this little thing here, we have 8 cc's left over. So is there excess tissue? Yes. Okay. If there isn't, obviously everything stops. If there's excess tissue left over, the next question you have to ask is it identifiable? If it is not identifiable, it is not a human subject because it is tamed for clinical care and it's not identifiable. And you can use this for research. Ken never knew. If it is identifiable, then it's now a human subject because remember, regardless of how it was obtained, it's identifiable. You have to have an IRB action, and the IRB will either say informed consent or they'll waive the requirement for informed consent before it can go into research. Many situations, let's say you're doing something very non-sensitive, so to speak, actually identifiable stuff can easily go into research with a waiver of consent, and again, Ken has no idea. On the research side, we have gotten blood, let's say from Ken this time. He signed an informed consent form for this. It's gone into research. The research is completed. There's blood left over. Do we need to go back to Ken for consent for the second research question we want to ask? Well, what the regs say is that if you strip all the identifiers, it is not a human subject because it's already existing. And again, so it doesn't meet those criteria. So we could use this for any other research without going back to him. On the other hand, if it's identifiable, you have to follow this through. So that the take home from this is that existing tissue, regardless of if it's existing from clinical care or from a primary research use, if it's not identifiable, not a human subject, don't have to worry about consent, don't have to worry about HIPAA. If it is identifiable, it's a human subject, and you got to do everything. So here is, because of these regulations, we are forced into this binary system. Human subject or not human subject, identifiable or not, okay? If it's not identifiable, this is the way our investigators see it. Open road. I'm on my own. See you later, Pearl. If it is identifiable, welcome to my world. Okay? So we have investigators who are falling over themselves trying to convince us that things are not identifiable. I would also. So, since identifiability is so important, how do we figure that out? I must admit, I think identifiability and privacy is overrated. Just people know who you are out there, whether or not you tell them. But here's what we'd have to do. First of all, there is no consistency between IRBs, and we are very proud of that. It's true. It always keeps those guys on their toes, so we continue on. So reality. Identifiability is a spectrum. It goes from identifiable to pretty much distorted, it could be perhaps to not very, to only LaTanya Sweeney knows. Now, how many know LaTanya Sweeney? Aha, very few. LaTanya Sweeney, many years ago, was at MIT, and it was a great thing. It was a hearing in the Massachusetts legislature, and the governor was up there, and they're talking about privacy and yada yada, and she just said, "Could I have your birth date and your zip code?" And clearly it was a guy, so she knew the sex, and right there in front of the whole, the hearing, on her little computer, she was able to pull up his medical records. The guy had some stuff that should not have been public, let me tell you. He's from Boston. But anyway, look at this. This is from 1990. If you have the zip code, the date of birth, and the gender, 87% of you are identifiable. Anyone here from North Dakota? You are identifiable just for living there. And so anyway, so you see it goes down. So it is hard to hide. The common rule, which is the one that tells us how IRBs have to work, allows judgment. Identity of the subject is or may readily be ascertained by somebody. So there's reality. Here is the common rule, which again, remember, makes us decide yes or no. IRB number 1 determination, IRB number 2 determination, and you're stuck with it. First one was us. We're very nice. Okay, HIPAA. HIPAA is actually a 4-letter word in many ways. This is a weird law. HIPAA defines identifiability by defining what is not de-identified. This is what we pay Congress to do. There are 2 ways of determining something is not identifiable. You can strip 18 HIPAA identifiers, or, right out of the law, you can find a person with appropriate knowledge of and experience with generally acceptable statistical and scientific principles and methods. Most people use the first one because we can't find that person. Okay? Now, the 18 HIPAA identifiers, many of them make sense. Your social security number, your medical record number. One of them, and this is just to make you all much broader in your education, is your vehicle identification number. Do you know on the front of your car, every time you take your car and you have it fixed, you know, some bozo comes out as you go in and says, "What's your vehicle identification number?" You go, "What the hell is that?" And there on the front of the car is this little brass plate that has about 40 numbers and letters. That's what it is. Why is that a HIPAA identifier? Well, you're going to find out. Well, when they were writing the HIPAA law, this is because of social behavioral research. They were doing studies on nefarious sexual practices at rest stops. And I'm sorry, guys, but it's only the males they were looking at. So what would happen is a car would come in. The research team was 3 people. One person would follow the guy into the John. One person was just kind of watching nervously with a cell phone with everybody's number. And the third would go over to the car and get the vehicle identification number so they could figure out where this person came from for their studies. That is why it's a HIPAA identifier, which is just phenomenal. But, so going back to my little cartoon here, that had totally nothing to do with anything, but I thought it was fun. So again, against the spectrum, HIPAA, identifiable or de-identified, and then because researchers went nuts with HIPAA, they put in this limited data set. Limited data set, you can have dates and you can have some geocodes. And you have to promise, I will not try to identify this person and I will not go back and ask for more information about this person. So it's a snapshot. If you want to do, sequential research on the same people, you cannot use a limited data set. But it does help for the snapshot stuff. Okay, the advance notice for proposed rulemaking. The common rule has been in existence for many decades, and people think this needs to be refreshed. So the way you refresh a law is to change it. And the process here is you first put out an advance notice saying, "Hey, everybody, I think I want to change the law, and here's how I want to change it." People have ability to comment. The people who wrote the new law can say, "Ooh, that sounds good, we're going to change what we do," or they can say, "Forget it, we're going with what we suggested." They then put out a notice of proposed rulemaking, again open for comment, and then they decide either forget it, bad idea, a different law, or what we suggested. 3 years ago, they put out an advance notice of proposed rulemaking. We have yet to hear what the feds think about this. I think I may be retired by the time they think. But what they do propose is significant for this topic. They have made this proposal that all tissue is identifiable. If one law says all tissue is identifiable, IRBs are going to have to consider all tissue identifiable, and it's a human subject. 60% of our current tissue research is done under a waiver, as non-identified, non-human subjects research. This will totally change tissue research. Okay, then you have genetics. And luckily, Holly said more about genetics than I've ever understood. Genetic information is unique, but is it enough to make a person identifiable? It depends on what kind of information you're talking about. It also, depends on if there's a reference set. And because I don't understand genetics very much, I go back to basics, NCI. So think fingerprints. I'm going to use Ben this time. So we come back from the break, and you find Ben up here, dead with a knife in his chest. and their fingerprints on it, okay? Ben wasn't really nice to me last night at dinner, so I took it very personally. Yeah, I'm now on my plane, or on my 5-hour taxi ride to the airport, trying to get my plane, and they're my fingerprints. Can anyone say Pearl did them off? Not unless I'm on file. Now, this may not be a surprise to you, but I worked in the post office in college. Hence makes going postal make sense. So you could find me, okay? So following that, we have a process which we call Crimson. Harvard, Crimson's a color, so it's kind of cute, so they call it Crimson. But remember all that excess blood from clinical care? Think about this river of excess blood going by. We have a process by which, in this example, an investigator can say, I would like 500 specimens of people who have asthma and are on steroids. So we look at the ICD-9 codes, we collect 500, we strip all the identifiers, and they're just labeled 1 to 500. Have a nice day. Not a human subject, yet it's enough information they can do the research on. This investigator says, I recognize 2 of the people. Can I go back and get more information? And it's because it all came from the same system. He had reference sets. So when people start talking about genetic information should be considered identifiable, it's really triangulating all of these things together. If you just find a drop of blood, no, it is not identifiable. Okay, now, so we go to our geneticists, and we have very smart geneticists. Just ask them. They will tell you readily, and they totally disagree. We have those who say, get a life, it is only theoretically identifiable, and those who say, really, it's going to be identifiable next week, so why not consider it identifiable? So this is what your IRB has to deal with. And remember, we are driven by a binary system of it is or it isn't. So be nice to your IRB members. Do participants care? No. They do not see the difference between identified or de-identified, and I think they've bitten in and swallowed the overpromise of genetics and thinks everything's identifiable. And there's an increasing push to, if not control, at least to know what's happened to their stuff. Maybe not only their stuff, but relative stuff. You have a brother who went and had genetic testing, has Huntington's mutation. What do you think your chances are? That's shared. You have an identical twin, same thing, as well as communities. So you got to really even go beyond just the individual into other areas. So now, how many people think they have participated in research? Every hand should go up. If it's still up, you've been napping or something there, because you are a research participant. So how do we consent people? Very poorly, very poorly. Okay, here's a broad research consent. By signing this consent, you agree to have your specimen placed in a research biorepository for future research on anything by anyone. Now, no IRB would let you do this, but we say the same thing, but in 10 pages, right? Okay, so I'm very trusting. I say, oh yeah, man, I'm really pro-research, so I signed it. Did I know that this blanket consent, they could do a full genome sequence, they could develop a chimera, which is mixing, animals, humans, making Stuart Little here. Creation, it would be a good Stuart Little. Let's not sneer at that. Creation of an immortalized cell line, or creation of a pluripotent cell. A pluripotent cell is a cell that can become any cell in the body, and what investigators are now doing, they can take a cell from you, push it backwards in the developmental stage, and theoretically make any cell at all, including a sperm. Okay? So when I signed this research consent, did I think of this? No, I was thinking cancer, diabetes, heart disease. Which of these should we include? You better get that phone. Okay. Then, here's the other one, and this goes back to the chart I showed you. By signing this consent, I agreed that my specimen will be used for diabetes research. Yes, diabetes is in my family. I want to support this research. I hate oncologists. I don't want my blood to ever go for an oncology study. And I think psychiatrists they're just not even physicians, let's face it. But did I know that by having, after the diabetes research is done, They could just strip my blood of identifiers, and lo and behold, it's going to go to every person I didn't want it to go to. This is okay by the regs. Most institutions will not allow this. At our institution, we go back to the initial consent, and if you consented for diabetes, we will not let it go downstream. But regulatory-wise, it is fine. Okay, let's look at clinical consents. I understand I'm about to have abdominal surgery for a horrible cancer. There are many risks, including, for example, death from anesthesia or the surgery itself. Then on page 3 in 8-font type is disposition of excess material. And actually, this paragraph is lifted from an academic institution. I understand that blood or other specimens removed, necessarily removed, may later be disposed of. These materials may also be used by the hospital or other academic or commercial entities for research, educational purposes, photography. You always have to add photography. I'm never sure why, but somebody likes that word in there. Or other activity, if in furtherance of the hospital's mission. By my signature, this is okay. Have I even read this? No. Okay? But we stand behind this. When you read in the front page of the Seattle Times that, your appendix became a sperm, we said, well, you said it was okay. Right. Okay. So what should be in an informed consent form? What is the research? What's a biobank? How the specimens will be stored? How they'll be used? By whom? For what? Potential benefits, any direct benefit to you? Well, I think you can argue that one. Is return of results a benefit or a risk? Depends. What is required of you? You got to give something, and access to data. So you really have to go through all the requirements in an informed consent form. The risks, tissue acquisition. Now, biobanking, I didn't biobank because I was afraid of the biopsy. We're talking a blood draw, get a life. Tissue acquisition, not really a big risk. Loss of privacy. That's one. And the way is it identifiable? My identifier be given to anyone who wants my tissue? Is the risk of privacy and potential discrimination higher with genetic research? I think most people would say yes, just because of the prognostication, like you are a carrier now for. Does the Genetic Information Non-Discrimination Act help? Yes, but there's still wide open doors. It does not cover all kinds of uses. Return of results, huge burden of information. Do you want it? Do you not want it? For yourself, your family members, your community? And I think the world is very much divided into different personality types. There's those who want to know and those who don't. I'm a not knower. I would not go to a palm reader for fear I'd find the one who knew something. I would never have my whole genome sequenced. Whereas, other people I respect immensely that you got to be kidding. Like, this is just good information you could act on. So I do think, it does come so down to a personal level. But no matter how much we talk about what should be in a consent form, remember, we often don't get one for all the reasons I talked to you about. The other issue with consent, we are now talking tens of thousands of participants. How long does consider if we wanted a consent form that went through creation of chimeras, creation of immortalized cell lines, pluripotent stem cells, that's a long consent. And just, logistically, how do you in fact do that? I really do not know. So, while this is still going on, think about other things that are happening in the public realm. And I'll just mention 3 particulars. The Immortal Life of Henrietta Lacks, which is written by Rebecca Skloot a number of years ago. How many have read that? Okay, very good. It's a big print. There are pictures. Those who didn't raise your hands, you got bad weather out there today. Go do something good for yourselves. The HeLa cell lines. When I was in medical school, we were told it was Helen Lane. It was actually Henrietta Lacks, and this was a woman who in the 50s, the regulations were written in the 70s. This is pre-regulations, okay? But she had what my mother would have called a female cancer. And she was being treated at Johns Hopkins, and they did extra biopsies. Let's say you needed 3 biopsies for your regular care. They did 5. And and it wasn't just it wasn't just Henrietta, but they were trying to create an immortalized cell line and figure cancer is growth gone amok. And if anything is going to be an immortalized cell line, that's where we should start. So she was the first one. Because of her cancer and likely comorbidities, her cell line took off like gangbusters and was the first truly immortalized cell line, which got big attention. She never knew about it, never permission for those excess things, no idea that her cells were going to be used. It was the 50s. There is some time warp here. But the way this was found out is one of her relatives, a few generations down, was sitting in a lecture hall, and someone was talking about the HeLa cell lines and explains who the patient was. And she goes, man, that sounds like my aunt. And goes up afterwards, and lo and behold, it was. And that just brought this, why didn't we know? And there are a lot of weird things that came out about this, but the bottom line was we should have known. Another one, neonatal blood spots. Every state, the kid's barely out, and you got to hurt them by hitting that heel. And this has really been, I think, huge public health importance. But what happens, you put the little blood there on those filter papers. Those are very easy to keep. It doesn't take up much room. You just dry them out. They're perfect. So what people have been doing is keeping those and then going back and saying, this is a great research resource. Think about if you had 1 million blood spots, and 4 years out, you could say, let's see how many kids got acute lymphoblastic leukemia, and let's go back and see if we can find a marker. Hugely important. But consent hadn't been obtained. So in 2 states, there were class action suits, and actually in Texas, they ended up incinerating a huge number of tissue, of filter papers, because families said, I would rather destroy this, not have access to this, than you violate my privacy rights. Now, most states are either getting rid of them and saying we're not going to do research, or getting consent. Okay? And the last one is the Havasupai Indian tribe and the Arizona State University. The Havasupai Indians live at the bottom of the Grand Canyon. It's a relatively small tribe. There is a lot of diabetes. And they actually had a very good relationship with the folks at the Arizona State University and said, listen, we've heard some Native American groups have a genetic marker for diabetes. Please come study us. So Arizona went in, and the tribe had a very high level of illiteracy, so the consent was oral, and there's been great difficulty finding exactly what that consent said. But what we do have is the consent did have the phrase to study the causes of behavioral medical disorders. It was broader than diabetes. The presumption of the tribe that this was only diabetes, and from here the story is almost identical to the Henrietta Lacks. The first member of the tribe to go to college is sitting in a lecture hall, and someone talks about migration theory using the Havasupai Indian tribe blood samples. And she goes, I gave blood for that, but it was for diabetes. And lo and behold, it was used for schizophrenia research. It was used to prove that that this group of Indians came over from Asia when the Bering Strait was frozen. They said we did not give permission for this. Now, is this okay by regulation? Yeah, they stripped the identifiers, and it was not a human subject. Unless, is it identifiable because of community? Okay? Bottom line, a lot of money changed hands, and basically the Indian tribe took all the specimens and gave them a proper burial. So, I do these 3 cases out there because they definitely are in the news. I think you need to understand it. And I think the whole concept that people do want at least to know, if not control, what happens. So I think even if the advance notice does not go through and we still can do a lot with de-identified specimens, I do think the expectation is very different than it was even 5 years ago. So where do we go from here? We could maintain the status quo, continue doing what we're doing, and just educate people. I think we talked about that a little bit yesterday. We could say, nope, you got to get obtained consent from every person whose tissue is used in research. The logistics of this are mind-boggling. There is also then the issue, do you allow people to say, well, you can do cancer research but not cardiology research? What if it's a heart tumor? I mean, as soon as you get into those little tiered things, I think they're really hard to figure out. Do you allow people to opt out? You can never use my specimen ever. How do you track that? And then today I opt out because I have no known diagnosis. Tomorrow I get diagnosed with a horrible disease and I'm very now interested in research. How often do you update it? So the logistics of that are difficult. I don't have the answer for this, but I think this is going to be one of the main things that we're going to have to spend a lot of time on. And then I'd just like to spend a few minutes on something that I think is a different problem with biobanking, and particularly the fact that I think biobanking has become synonymous with genetic research. I think we are suffering from what I would call overpromise of genetics, as well as, what I mean here by collision with clinical care, I think genetics in the research environment is way far ahead of genetics in the clinical space. And I think some people are actually going into research because they think, well, my doctor's not going to do it for me, I'll go into research. And I think there is a collision here. So in terms of the overpromise, these are some phrases that I pulled out from some of our research proposals. I'm not saying these were approved, but we wonder why people are confused. Genes are what determine who you are. Okay, that's even bad English. Most diseases have a genetic basis, or at least whether or not you have a predisposition. With genetic research, we shall be able to identify diseases before they occur and possibly prevent or delay them. Now, you can see where the intent came, but if you know nothing about genetics, you can see why this is really quite promising. And then you have the direct-to-consumer, genetic product. Well, man, if I have to pay for it, it must mean that this is valid information. If you look on any of those websites, and if you're ever in a really boring conference call, which I am a lot, I would really suggest surfing for consumer genetics, like direct-to-consumer. There are an incredible number out there. 23andMe is obviously one of them. But there is this other one that is really kind of fun. It's essentially defining paternity, okay? And you open up their website, and there's this cute little child who has this very quizzical look on his face and a little smile. And the title says, "Add spice to your family reunion." So what you do is you nefariously get samples of, whoever you think you're related to. All the mailings are done in plain wrappers, so no one knows what it is, and you can find out. Now, as I'm saying this, in the U.S., depending on which geneticist to whom you speak, 12 to 15% of you are sending Father's Day cards to the wrong guy. So this is not a small issue, okay? Anyway, I know mine is mine, so your numbers have just gone up. Further over, this one I love. It is like reading a book. That's not a book I would buy. And then everybody goes back to the Mendelians. If you have the Huntington's mutation, it's hard to argue what's going to happen there. And these multifactorial variants, et cetera, it's hard to even begin to get me to understand this, let alone the 10,000 people I'm going to bring into my research study. So this is very difficult. But I do think there is this concept that genetics, if it is there, it means I have the disease. Carrier status, some may understand that, but I do think there is an oversimplification, overpromise. Then there is the availability. I think their concept is it is more available in the research realm than in the clinical realm. And I think there are huge challenges to this. I think there's a lot of problems of educating clinicians who are not genetic specialists to understand what is going on. And you think about regular diagnostic testing. You usually have an idea, like, a person comes in and their urine output is really weighed down and they're feeling kind of bad. You're worried about the kidneys. You're going to order a BUN and creatinine. They have a juicy cough. You're going to get a chest x-ray. So you are focused. You're targeted in what you're asking for. And there's usually sort of a negative positive that you're somewhat comfortable with. Within genetics, particularly as you go to either whole exome or whole genome sequencing, there's very little limit on scope, if any limit. And then you get the results back. And I love this. It could be positive, uninformative, negative. Well, you don't have the mutation, but we don't know what that means. Truly negative, variants of uncertain significance, and, as was said, as Holly said, check back next week, next year, next decade, because now we might find that that's really a bad thing to have, but today we think it's okay. Go have lunch. So genetic results, I think for clinicians, are extraordinarily difficult if you're not a geneticist. Another area that I found intriguing was, do regular clinicians have not only the expertise but the time? How are they dealing with genetic findings? General practitioners, when you go to see your PCP, you're going to be there about 20 minutes. That includes the weight, the blood pressure, getting undressed, having the exam, talking to the physician, and getting dressed and paying the bill. Not a lot of time to talk about that variant of unknown significance, okay? There's a paucity of genetic counselors. Who do you go to for help? And genetic counselors are way underpaid. And this last one, this study is a little bit old. I hope they're doing better. But look at this. 86% of people in a primary care practice said that when they did do a genetic test, they called the patient by phone to tell them the results, and in most cases, it was the office secretary who called. Hey, that test we did, it's positive. If you have any questions, call us back. Nah, not appropriate. Here's also interesting. How do patients feel that their clinicians are ready or not ready for helping with genetics? They looked at people who had done direct-to-consumer genetic testing, and you can see there the vast majority said that they thought they would share the results with their doctor, but in fact, very few did. And then when they asked, why did you share them with their doctor, a different study, about a quarter said they thought their doctor could help with the interpretation. Only 25%. Look at that last one. I gave my results back because I'd like my doctor to realize that genomic medicine is actually starting to be useful. Okay? You have just flipped. You know, patients are saying, you got to start getting in the game. And the reason I put this in here is that we are seeing, and I think this is driving a lot of the return of research results. You know, as it's research, is it ready for prime time? Yet I can't get it in prime time, so I really want to do it. So hopefully in the question and answer period, we can go into that in a little bit more detail. Return of research results, thank you, Holly. But I want to tell you two stories on return of research results. And the first one is my personal one. And this is actually, don't worry, I'm not going to die up here. But this was actually a game that was played at a meeting. It was a dinner after the meeting. We're sitting at a table with about 10 people. And it was actually Karen Rothenberg who was the speaker. And on your dish was an envelope and it said, do not open. So, you know, I wonder what this is. You're doing this, you know. The nice people just sat there, but I'm like, and anyway, so she gets up and she speaks. She said, okay, I want you each to open that envelope, and inside that is another envelope. And on the front of that, you know, as soon as you open it up, it'll tell you what disease runs in your family. And for this exercise, there were only 4 diseases. There was Huntington's, there was hypercholesterolemia, Fragile X, and diabetes, okay? So I opened mine up, and I have Huntington's. She said, if you want to know your result, open the envelope, the next one. Everybody who had hypercholesterol, you know, they're all like, I sat there, I thought, Huntington's, do I want to know my result? Well, I finally opened it because I wanted to make sure it was negative, and it was positive. And I just wanted to stuff that puppy back in there. And this was an after-dinner theater. I cannot imagine how people deal with information. You know, you go to the doctor wanting to be told you're fine. You're not going there for, ooh, you're fine, except for that liver nodule, which might be cancer. I mean, you know, it's like you're not prepared for that. And I think in genetics, it's stuff you hadn't even thought about. And then this was a study that was being done. It was an example, and I kind of changed some of the details. But it was a study looking at effect of getting genetic results. And in the study, they were looking at 20 common variants. And as you came in, you could say, I only want to get these 2, or you could get all 20. And ApoE was one of them. And with ApoE, if you get a double hit, your possibility of getting Alzheimer's disease is increased by somewhere 10 to 15 times, not percent, but times. So Ms. Green, her mother and 2 maternal aunts died of Alzheimer's disease, and she was 50. And she wanted to know if she was at high risk, because she needed that information for life planning. She came back with a double hit of ApoE and essentially planned in a very harsh way. She'd said, you know, she would plan a suicide once she got symptomatic. But for her, it was empowering. Mr. Blue worked in an office, and his desk mate says, hey, there's this study. You got some diabetes. Why don't you come? This is kind of fun, you know, genetics. This is great. So, okay, so he goes, yeah, and he, you know, he's healthy. He marks, give me everything back because I'm just healthy and I'm just doing this for fun. He gets a double hit of ApoE, and it devastated him. He lost his job, he lost his wife. It's like, this is all I can think about is I'm now getting Alzheimer's. Same information, very different impacts. And once it's out there, you can't put it back in. So, to finish, final comments, tissue is needed for research. A lot of tissue is needed for research. I think genetics highlights challenges that we've never really gotten our arms around, such as identifiability, who is a subject when you're talking about communities, return of research results, informed consent, and I think the public needs and expectations are definitely changing and absolutely need to be addressed. So I see it, and this year with all the horrible weather, I must admit this slide is becoming more real. So the perfect storm, we have genetic risk, identifiability, secondary use, return of results, and these are coalescing. And in that little building is your IRB office. It's true. So I will stop there, and thank you very much.
All right, so we're going to bring Holly back up, and joining us on the panel is Ben Wilfond, who was up here much of yesterday but has not been formally introduced, and I think most of you know that Ben is the director of our Truman Katz Center for Pediatric Bioethics, as well as an attending physician in the pulmonary division at Seattle Children's and professor in chief of the Division of Bioethics. And Ben came to us about 8 years ago from the NIH, where he was part of the Social and Behavioral Research branch at the National Human Genome Research Institute, as well as the Department of Bioethics in the clinical center. And Ben has done a great deal of work in genetics as well as research ethics. So he's going to join the panel. And we've got microphones in the aisles, and we're open for business. So if people have comments and questions, we'll get started. Yes, right here.
Hi, thank you all. I am thrilled, that was great. I'm in Russia, Columbia, and I'm the ethicist for we do clinical whole exome sequencing. One question and two comments. I'm curious, Holly, why you would call the primary, why your group would call the primary ciliary dyskinesia an incidental or secondary finding when that was one of the symptoms. I understand it was in a research context and you were probably narrowing down the target, but that was one of the symptoms. And then the two comments, and if you can, the two of you can make some statement about it, was I think that one of the other issues that was elucidated but not gotten into is both for the geneticists as well as for the patients and families, the extreme variability of even when we have a diagnosis, even if it's not uncertain significance, when we know what it is, whether it's cystic fibrosis or another disorder, we really don't know there's such variability of expression. We don't know what to really tell them, what to expect, oftentimes, certainly not all the times. So I'd love to hear you talk about that because it's true for the return results. That's such an important component.
I can repeat.
You want to repeat it? Yeah.
Yeah, so the first question was why we would call primary ciliary dyskinesia a secondary result if it was one of the symptoms. So the objective, going back to Pearl's description of informed consent forms, the objective of the study was to identify the gene for Miller syndrome, and which is a limb malformation disorder. One of the very good things about exome sequencing is that a lot of rare conditions the genes are not readily identifiable using other genetic research techniques like linkage analysis and association analysis. And there were at the time about 30 families in the world identified with Miller syndrome, and this was the only one that had any lung symptoms. And so it was honestly not known if this was a manifestation, but we were not looking for a diagnosis or an explanation of their symptoms. They had a phenotypic diagnosis of Miller syndrome on the basis of their limb presentation. So it was in fact really secondary in that capacity, but it was in fact identified as a result of looking for the gene for Miller syndrome, not as a result of looking at a preset list of secondary results, which is sort of what I was talking about later. But I think going back to your other question and partly responding to Pearl's excellent talk, I don't know, is that slide at the end supposed to be you and me walking away? No, I meant me. I was thinking, yeah, I felt bad. But I think I'm more. Oh no, that was not my intention. I am more optimistic about the potential for genomics in affecting medicine in people's lives. And I think it comes partly from the interviews with the families that I talked about, and it comes partly from my recent experiences over the last 8 years with families who are actually in clinical genetics settings, and families who don't have diagnoses and have often very sick kids and are looking for explanations for that. And I think, or families that have kids with chronic disabilities and even adults who have sort of conditions without clear explanations. I totally agree with you that people have very different perspectives on things that are filtered through their values, and we need to really think through that. And when we think about what to offer and how to explain things and what the potential risks and harms might be. But I think there are people who have very different perspectives of even what an uncertain finding might be than what you or I might have from our perspectives of our life experiences. So I want to know more about what different populations think, and I want to study more about what they do with that information. There's an article in the New Yorker this week by Seth Mnookin about a family with a rare genetic condition who was part of a research study where that was identified and other families were connected, and I think that's a very insightful story about how some families experience it. And you may say it's just families with rare disorders, but when you look at the number of people with rare conditions and even undiagnosed rare conditions in the world, it's actually more people than you think. So it may or may not be that useful for diabetes or even age-related macular degeneration like I talked about, but for some people I think it's going to be very, very powerful and very, very important.
That's actually not what I was saying.
Yeah, I know.
I'm fully in agreement with that. I'm probably one of the biggest proponents of that. What I'm talking about is how do we, in returning results, and it gets probably back to the informed consent process rather than just the form, is the idea of explaining the variabilities.
Yeah. And
because
I think that's
for the rare diseases, as well as somewhat more common, but particularly the rare diseases, I think we just have to make that for the physicians as well as the patients, the understanding that we can't always decide be able to explain whether it's an adult onset or whether it's a childhood onset. Doesn't mean not doing it, and there's huge value in it.
And I agree with Pearl about some of those issues. Maybe you want to comment.
I think that's no different in genetics than in general medicine. I mean, I think it's exaggerated just by the number of potential findings, but I think, clinicians deal all the time with, here's the basic diagnosis, what exactly your history, what your course is going to be, we're going to have to follow. So I just think we need to maximize those tools, in a broader way. I don't think it's different.
And I would assume you'd both agree that context is important, that there may be differences in the way at least some people feel about having genetic testing when they're symptomatic versus just sort of general screening for...
Well, and I think, as Pearl said, and I agree, there's a lot of examples of physicians who don't have training in genetics offering and returning genetic results when they don't completely understand it. And so I think there's definitely a lot of important things that can be done to minimize that. There's a lot of literature about ordering the wrong one. So I agree.
Steve, I think you're next.
Thanks both for terrific talks. Question primarily for Pearl, although anybody's welcome to take it up. Pearl, you briefly mentioned GINA and the protection against discrimination that it offers, at least in health insurance and employment. I wonder if you could speak to the extent to which, at least in the health insurance realm, the Affordable Care Act provides additional protections that may even go beyond GINA in terms of prohibitions against discrimination based on pre-existing conditions and that sort of thing.
Other than saying, I agree, but I don't think, I'm not comfortable saying exactly concretely how much better those protections are. I think we're going to have to see how that plays out in the marketplace. I think the whole insurer concept of how they're dealing with genetics in general is so difficult to figure out. I mean, we have a lot of the whole quote theranostics of, a lot are like for pharmacogenomics. Are you a rapid metabolizer or a slow metabolizer? Insurance companies should be mandating these before some of these drugs are prescribed, yet very few are. So I think exactly how they're going to incorporate genetic information into any of their decisions, I think will be interesting. I do think you're right, though, that there is going to be some added protection, at least for health. I mean, the issue is, life insurance. Haven't seen that one. And we're a discriminating society. We discriminate on, age, sex, et cetera. We have laws against that. And you see how well we do with that. God, I'm negative today, aren't I? It was all that traffic talk.
Yeah, it must be. Go ahead.
Hi, I was wondering about whose responsibility you think it is to follow up on those variants of questionable significance when they come back. We had a family that came in for genetic counseling after a very severe phenotype of a child was born and had one of these variants and no one knew what to do with, and they went ahead and had another child thinking that it was not genetic, and the next child had the same phenotype. And when they went back and actually talked to the lab that had done the original testing, they said, oh yeah, we did find this 6 months ago, that it looked like this probably was causing this phenotype, but the lab didn't call, and the genetic counselor doesn't have the time to call back on every single variant that comes without kind of speaking to the family.
That is a huge issue. I don't know the answer to it. I don't think that medical charting and kind of maintenance and gathering of medical information is even beginning to be ready for this. I mean, how much genetic information, if you do a whole exome sequence, is that raw data going to be in your medical chart? Do we even have the IT infrastructure for doing that? You can just wait a minute there. I see you gasping for air. And you're right, it was you. You were the bigger one. Anyway, and I think there's a lot of that infrastructure that needs to come, but you're absolutely right. I mean, some of there's a newborn screening study that's coming out, and their plan there is just to use that information. I think it's a limited time period, I think 5 years out, I think it is. Then who's going to paw through that? So I think we definitely, it is your genome sequence. If there's the reading today may be different, not only for the one unknown significance you found, but there might be other ones, 2 years out. And I don't think we have the process for dealing with it.
Go ahead, Ben.
I just want to add that the point that you're asking about is actually not unique to genetics. This is true for clinical medicine in general, that anytime a person has a clinical encounter, they gather information about that person and make a recommendation. Facts change constantly, information changes constantly, and you can ask the same question: what obligations do clinicians have to go back to patients they've seen 1, 2, 3, 5, and 10 years ago, because now we know more about the treatment of asthma than we knew then, and the recommendations we gave then were no longer correct.
That is so smart.
Laney?
So a comment and a question. So first, I just want to point out, I enjoyed both talks. Pearl, you began by saying you think we've overhyped genetics, which I actually agree with. But we've also hyped genetic determinism. And your example of Mr. Blue and Mr. Green was a perfect example of that. Having 2 copies of the ApoE4 does not tell you you're going to get Alzheimer's. It's just going to tell you that you're at increased risk. And so for the guy who was devastated, I'm really concerned that he didn't really get good counseling, because then he would realize that his parents and his grandparents and everyone who knows who never suffered from any of this really could have had it. And it's on that note that my question is actually directed at Holly. So, we have these 56 genes that we're told are that they're highly likely to express. We know that because we've studied them in high-risk populations. We actually don't know what they mean in the low-risk general population. And so when you tell me that 4% and 0.8% of the Ashkenazi and non-Ashkenazi Jewish community has these, that may not necessarily, unless there's family history, actually correlate with significance. And how are we going to really try to explain that as we move into the general population?
That's a really good question. So actually in that paper from ClinSeq, they actually did look at family history and look at whether there was, was it really potentially a risk factor in the context of other factors or not? And in some families it was, but it wouldn't have been picked up using standard algorithms. So it's a really interesting question. In the case of long QT, there was another paper I can't remember the name of the author right now, and there was another one on dilated cardiomyopathy where they looked at known genes and known variants and basically raised exactly the point that you're making, which is that in fact maybe they aren't as highly penetrant as we think they are, and therefore we don't know what to tell people. I think the argument, which resonates with me a little bit even as I has concerns, is that for something that you can do something about that has such an adverse consequence like lung QT or dilated cardiomyopathy, that at least giving people the opportunity to know that gives them the opportunity to take action. But I think I agree with you that if we don't really know, we may be causing people undue stress, and many people, as Pearl said, will not, will misunderstand and will take it very personally, and it may in fact ruin their lives. And so I don't take that risk lightly. But I will say that I had a friend who died of sudden cardiac death this fall, and I know that who had a family history of it and didn't know if she had never had genetic testing, and I think that if she had had the opportunity to know, even if she wasn't sure, the person who she was with her values and her perspective, which isn't for everybody, would have wanted that opportunity. And so I think it's, as with most of the things many of us here think about, it's weighing the benefits and the risks and thinking about the best way to proceed. But I think it's a very good point.
I'm really confused by what you just said, because you made 2 points that contradict each other. One is if she had the opportunity. She had the opportunity. If she went to a cardiologist in the last 2 years, they could have done, even if they didn't do whole genome sequencing, they could have done a gene chip. Mm-hmm. And your second example, which was, I think people might want this information to act on it. They may be acting inappropriately because they're really not at risk, and we're exposing them to a lot of iatrogenic harms.
Yeah.
I mean, putting in a pacemaker because I'm worried that I have long QT when I don't, I could do damage to myself.
Well, yeah, so to clarify what I was saying, I think, so first of all, I think the magnitude of the iatrogenic harms is a really important empirical question. How often pacemakers would actually be put in people who don't need it and don't have family histories, who aren't examined by a cardiologist, I don't know what that answer would be, and I think that would be important. I think my point about my friend was that she had not perceived that she had a family risk, and so she did not go to a cardiologist, and she did not get her primary care physician. I don't know all the details, but for whatever reason, she was not presented with that opportunity. So would she have taken up exome or whole genome sequencing or a cardiac panel if it had been offered to her? I don't know, but I do know that many people, going back to the ACMG, the part of the ACMG guidelines that I agree with, I think that there are people who would like to have the opportunity to think about what they would do about that information. They may or may not choose to do anything with it, and they may or may not do the right thing, but as Ben said, I'm not sure that's unique to genetic information. I think people do that with health information already all the time.
Ben, you want to...
I just make a really quick point. I just want to remind everyone that the most common genetic test is a family history, and Holly's example was simply that very basic genetic test is what was not done effectively in identifying her risk, and it's not and the next step would have been going to see a cardiologist for clinical testing, not necessarily even genomic testing.
And it may also be worth pointing out that these issues get even more interesting in the research context. In other words, when you're not doing, now a lot of the research Holly's talked about is actually with people who have identifiable traits. But when we start talking about doing research on a general population, for example, and then returning results to people who maybe were not expecting that something is wrong, obviously the issues get really interesting. Yeah.
Hi, thank you for these talks. They were really illuminating. My question is that not all families come about in the typical way, so I'm just wondering if sperm banks, for instance, are going to have an obligation to do whole genome sequencing on those sperm that come in there. I have friends that have 2 kids with hypertrophic cardiomyopathy. The donor didn't know when he donated the sperm, come to find out later when a baby from another family died. Obviously, they wouldn't have picked that person if they had known. So what are your thoughts on that?
Yeah, go ahead.
I do think that already there is genetic testing with regards to reproductive planning and sperm donation, and I think that I think it would make a lot of logical sense in that context. If your reason is because of a particular condition to have that testing for those things, so I think that's actually a great example where genomic testing could be very useful.
Yeah, I mean, I think as a standard screening test, it's probably too expensive for most sperm banks to offer as a standard thing. However, I think it will be used. There are some papers, including one I was involved in using whole genome sequencing for non-invasive fetal genome sequencing early in pregnancy to identify things that you might want to identify for screening later. I think the other context that we've done research on is we've talked to parents of adopted children about what genetic risk factors they wish they knew about their children. Again, more focusing on fairly bad things that they might be able to do something to prevent when they don't have any family history. But I think there are also important implications and risks of, how that information could be misused and also could deny adopted children, the right to make their own decisions. So I'm again saying an inherent contradiction, but I think there are pros and cons and benefits and risks, and that's part of what will have to be decided as we implement policy.
And I would add, God, this could be a good TV movie. I agree that you'd want to have a sperm that is a good guy, doesn't have any of the bad things, but we are so limited in our analysis of, whole exome sequencing or whole genome sequencing. You would have to start worrying about what is the liability of the sperm bank for, that was a 10-year-old sperm. How come, when the testing was done, it was this, as well as the implications for the donors. The entry in is having to know all this stuff. I'm sorry.
I was just going to say, this has happened. I'm probably going to get the details slightly wrong, but if you come up afterwards, I'll find the reference. There was a sperm donor who donated and has several dozen children resulting from his donation who had a long QT, I think it was long QT, one of the cardiac genetic conditions and didn't know it, and a large proportion of the children now have those conditions.
Yeah, these are my friends.
Is that your friends? Oh, okay.
Yeah.
There you go. So it's
Strangely,
not long QT.
yeah.
So yeah, and it's a large number. It's not just your friends. It's a fairly large number.
20-something.
Yeah. So I think it's obviously it is a TV movie. Yeah. It's, and hard on those families, definitely.
Thank you.
Yes.
So can the speakers address the issue of how, given the uncertainty of certain genetic findings through these new techniques, may lead to additional risk for children, particularly with additional interventions and evaluations that people may do? And are the pressures there different than in the medical decision field when there is uncertainty also? How I as a physician may handle that in terms of doing additional testing on my patient or not? So can you address those two scenarios, if they're the same or they're not the same?
So I think that's a really good question. I will say from my research and also from my experience on the Clinical Exome Sequencing Committee at Seattle Children's, that I would say the majority of people who are being offered exome sequencing in a clinical setting now, and a very large proportion of those who are probably being sequenced in a research setting right now, are people who are often unsure of their diagnosis and have undergone substantial diagnostic odysseys. So in fact, they are already undergoing a lot of procedures and tests in an effort to find a diagnosis, in an effort to treat and improve the condition of their child in the case of children. And so I think the argument has been made that perhaps identifying the condition would actually reduce the number of extra tests and risks that they would have to undergo rather than increasing them. At least for those children in that situation. Now, I think if exome sequencing expands to a broader population, I think the point is very well taken that the iatrogenic risks of other procedures may in fact be greater. So I think that's a very valid point. But at least in these kids, and again, the New Yorker story talks about this. There's some other videos online. These parents have spent an enormous amount of money and time and anguish going to institutions all over the country sometimes trying to figure out what's wrong with their kids and having lots of procedures. So
That may be arguable for the primary case, but as you know, we have siblings, and so there may be pressures to get the siblings to undergo additional medical testing that otherwise we wouldn't do. Is that scenario possible?
Well, I'm trying to think about the examples that I know. I suppose if it was a test that was predictive rather than diagnostic and the siblings didn't actually have conditions, then I think that's possible. Most of the situations I am currently aware of, if the siblings either have or don't have some of the symptoms, and so they would only undergo those tests and procedures if they actually seem to be developing the same condition as their other sibling. But it's a good question. I have to think about it.
I would add, my concern is also in the other direction. I fear there could be misdiagnosis. The presumption that, oh, we know why this kid is wheezing because he has such and such the cilia don't work. When in fact the kid has basic asthma. I have a little bit of concern, like you think you know the answer. And the other issue is self-fulfilling prophecies, particularly with behavioral stuff. Like, your kid has a marker at age 2 months of hyperactivity. I think that kid's going to be hyperactive. Yeah. I mean, so there's a little bit of that. So I think it's really a two-way street here. And I think we really need, I think we're going to be in a totally different place in 10 years. We're going to know more. It's going to be a more accepted standard of how to approach this stuff. We're going to have ways of maintaining the information through time and being able to go back, I think, and reference it and update the analysis as opposed to, updating the test itself. It's not like, any other test in medicine, you just repeat it. These should not be repeatable tests, hopefully.
Thank you.
So I think we have time for one more. So Rosemary, you want to ask the last question?
Right. I have a question about the reconsent of children when they become adults. In the IRBs, very routinely, we waive the requirement of reconsent of the children for repositories when the tissue repository has been authorized by the parents. And what I'm wondering is, what are your thoughts about that? What is the ethical thing to do? Because it's very difficult to go back and to ask the reconsent, but is it the right thing to do, to waive?
So once a subject has turned 18 and they've got materials in a data repository, what do we do about consent?
What we have started doing is, and we hopefully get smarter every week with this, when it's a child who's in a tissue bank, we say that we will try to recontact that child at the age of majority to have him or her agree to whether or not they want to continue to have their stuff in the bank. We then say if we're unable to reach you, what we'll do is de-identify it. We don't get rid of it, but we de-identify it and it goes into into that sort of use category. But this is coming from a bank where stuff is kept identifiable with updated information. But if you can find the person, they say, no, I want it out, it's destroyed.
Yeah, I think it's a real challenge to researchers because it is hard. What I've seen happen is that IRBs frequently require the researcher to make a very good faith effort to contact the child, now adult. But then either do what Pearl described or just say, well, we made our good faith effort and that's good enough. I will say, just to reassure everybody, because I think the identifiability is an important challenge and the risks associated with it, but having spent a ridiculous amount of time and having some looking at thousands of consent forms for dbGaP over the last couple of years, it's really, really hard to get data from dbGaP at least, which is where a lot of the research sequencing data has gone. Putting it in the cloud, I think, is a lot easier, but I think getting it from dbGaP is really, 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 really hard. So hard that no sane person would really want to try it. So the good news is, perhaps, that the risks of anyone using it for nefarious purposes, accidentally or intentionally, is rather low, I think, at the moment at least. So.
And Ben, the last word.
Yeah, I want to respond to that question, then make another final comment. But about 10 years ago, we did a general population study that had two questions that are relevant to the comment. One is the point that Pearl made, that very little of the public cares about this distinction of identifiability or non-identifiability. Our sample, only 16%, tracked the federal regulations. We also, though, when we asked the question about what about if your parents had given a sample and they're now 18 and they can't find you, would you care about this? Only 25% it didn't really bother them that this might be going on. And so I actually think that as an approach, I totally agree with the idea of trying to recontact people, but if you can't reach them, to me, if there's scientific value in keeping those identifiers, it's not clear that there's any value in de-identifying it. The last point I want to make really quickly is, in hearing both Pearl and Holly's talks, which I thought were both excellent, I think one of the challenges is that there's a tendency, in fact, I've actually used the metaphor from Winnie the Pooh of Tigger and Eeyore, that Tigger, everything's going to be absolutely wonderful, and Eeyore, everything, the sky is falling, it's going to be terrible. And then there's Christopher Robin, who is very deliberative, very contemplative, plodding along, and I think that's where genetics is going to be. It's not going to be the panacea. It's also, though, not going to be a lot of the things that Pearl's worried about. And I think we will figure out how to integrate this.
So, Pearl, I think he just called you Eeyore, but please join me in thanking our speakers and panelists. So just a couple of important announcements before we go to break. If you plan on leaving the building and coming back, please remember to bring your name badge. There is a book table out there, and they are selling The Immortal Life of Henrietta Lacks, if anybody has not read that and wants to do that today. Also, don't forget about signing up for CME, and if you need parking, you can get that validation too. And while you're out there, please thank the staff. This conference, part of the reason this conference runs flawlessly is that we have a staff that just puts in an enormous amount of work to make sure that your experience is a good one. And there are too many people to name, but it starts with the planning and obviously our center faculty, as well as people we ask to serve on the planning committee, help us identify speakers and topics. Ben Wilfond is extraordinarily helpful as I try to move forward and inviting speakers and putting the program together. Rich DeLine does a marvelous job with the AV and makes this run seamlessly. And when you're out at the table, give special thanks to Kathy Fennell and Kathy Cohorn and Kate Landry, who spend probably hundreds of hours getting ready for this conference every year, along with the rest of our staff who's been helping them, and you'll see out there. So give them a thank you when you see them. So we've got about a 15-minute break at 11:45, at 11:30. I'm sorry, 10:30. You're right. Okay, good. 10:30. You're going to go to breakout sessions. There are 4 of those, and you want to refer to this sheet in your syllabuses that will tell you what the 4 are. They are all going to be excellent. I have to admit I'm not entirely sure which one I'm going to go to because all 4 of them look terrific, and they're all very different. Hopefully there's going to be something there for everyone. The rooms are... one of the first session will be here, and then the rooms are sort of 1, 2, 3, 4. There will be a label that correlates with the number on this sheet. And they basically, 2 is the closest room to us, then 3 is a little further, and 4 is a little further, and the staff will help get you where you want to go if you can't find the room. But take a look at the syllabus to see what you want to go to and enjoy those breakouts, and then we'll come back here at 11:30 for our concluding talk by Alex Capron.
Hi, guys. We're actually going to go ahead and start the next breakout session that's in this room. And I know it's a pain because you have lots of stuff with you, but if folks could come down closer, we're hoping to have some discussion around this topic. And if it's not too much trouble to come down a little closer and make it a little more conducive for the speaker to interact with you. I'm Maureen Kelly. I'm one of the faculty members at the Center for Truman Cats and Pediatric Bioethics. And this topic for this session is actually really interesting. It's looking at the issue and a kind of pressing issue in public health surrounding creating countermeasures for children in the event of a public health emergency, but in particular in the event of a biological, chemical, nuclear, radiological attack. And so there's some really interesting ethical issues surrounding, well, how do you prepare for that, right? If we don't know what countermeasures would be effective in children, don't we have to do research? Internationally, WHO has been looking at this question, and we're really fortunate to have Michelle Groman here from the Presidential Commission that has come up with a very detailed report, a very thoughtful report on this set of ethical questions, and have made recommendations about what ought to be done ethically to prepare for countermeasures for children. So let me just tell you a little bit about Michelle. She's currently Associate Director at the Presidential Commission for the Study of Bioethical Issues. She's been involved in a number of important reports at the Commission. The first was Gray Matters, Integrative Approaches for Neuroscience, Ethics, and Society. The other is the one you'll be hearing about today. And a third is Moral Science, Protecting Participants in Human Subjects Research. And prior to joining the commission, she worked as a practicing lawyer with Jenner and Block as a litigation associate, and she also clerked with a judge. And she graduated from Harvard in biology and then received her JD also from Harvard Law School. So really pleased to have her here and to have her expertise on this interesting question. Michelle?
So thank you very much, and thank you to all of you for being part of this breakout session and hopefully a little bit of discussion. I know the room is a little bit spread out and we have microphones around, but I do hope we'll have a little bit of time for interaction at the end. And also just thank you to everyone who's put together this conference and organized it and invited me and the Commission to take part, because this really is a report that we hope to discuss in more depth with really relevant stakeholder communities, and clearly this community of pediatric researchers is one that we certainly want to involve in getting the word out about these recommendations, but also hearing some feedback about them. And that's kind of the initial caveat I want to make to this talk, or maybe initial ask, which is as I go through and talk about the Commission's work, what I hope we can discuss, and I'll offer some commentary throughout, is how these particular recommendations, the Commission's analysis in this area, can apply more broadly. We'll be talking about the regs, the preview that will really be focusing on Section 407. So there's a lot to be said about Section 407 research more broadly, but also perhaps pediatric research more broadly, and to keep that in mind as we talk through these recommendations and analysis. So the other opening piece I just want to offer is that medical countermeasure research really challenges traditional research ethics, and in that way is very much, I think, at the core of what this conference's theme is all about, which is the boundaries of ethical research. And the Commission put in a lot of time, a lot of thought to come up with a careful way of moving forward ethically with this research, and I hope that you can appreciate the challenge that was before them. I think our chair said at the time that it was the most challenging report they had worked on to date, and I think it's still very much true. So to get started, I really will go just very briefly to introduce the Commission on whose behalf I'm here speaking with you today. They were formed in 2009. Sometimes people don't realize that the bioethics commissions on this line of national bioethics commissions have all been temporary and generally have gone up and down with administrations. So this is established by executive order by President Barack Obama in 2009 to look at a wide range of issues to ensure that medicine, science, technology move forward in an ethically responsible manner. This is a picture of our current commission. They were 13 members at the time of this report. They are now 11, as you can see here. 2 have since retired from the commission. Our chair is Dr. Amy Gutman. She's the president of the University of Pennsylvania. Our vice chair is Dr. Jim Wagner. He's the president at Emory University. Some of these names might well be familiar. They really do represent a varied range of expertise, much like the field of bioethics itself is interdisciplinary, so is our commission. Experts in philosophy, law, medicine, bioethics, clinical research, engineering, theology, really across the spectrum of relevant expertise for a commission that deals with issues as broad as the ones this does. So since their inception, they've published 7 reports. They're quite prolific, at least by my accounting. They started with the first one you see here on synthetic biology, moved on to doing a historical review and ethical analysis of the U.S. Public Health Service research, STD research in Guatemala in the 1940s. They took a look at current human subjects protections, also a report on privacy and whole genome sequencing. We then have the one we'll be talking about today on pediatric medical countermeasure research, another recent report on incidental findings, and finally the most recent, which just came out in May, on neuroscience and related ethical issues. And that's a charge they received from President Obama about a year ago now as part of his BRAIN initiative, and that's the project they're currently still working on. The first report, Gray Matters, was a volume 1, and volume 2 is where the commission is now focusing its efforts. These are all, just so you know, available on bioethics.gov. There's a lot of material there if you're interested in what the commission's doing. In addition to these reports, for example, the commission has been very committed to developing educational materials to make these reports more accessible for educational settings of all levels, traditional and non-traditional. There's really quite a bit there, and I'm happy to chat more about it for folks who are interested afterwards. So let me get into the Safeguarding Children report and really the events leading up to it. So it really came to the fore in early 2011 when the government undertook a preparedness exercise simulating an anthrax attack in a city like San Francisco. What they found is that about 8 million people would be affected, and nearly 2 million of them were children. And while plans were in place for what to do with adults, there wasn't such a path forward with children. The current plan is to provide a 60-day course of antibiotics as well as AVA, which is anthrax vaccine adsorbed. So the anthrax vaccine as post-exposure prophylaxis. But what quickly became apparent is that there are no data about the use of AVA with children. So even though it would be provided in an emergency, the question is, well, is that really what should be done when there's no data on the safety, immunogenicity, et cetera, of this particular vaccine? So after that exercise, the Assistant Secretary for Preparedness and Response asked a different federal advisory commission to really look at the science and security issues surrounding this, asking what should be done to prepare for an anthrax attack and how to treat children in such an event. That board, the National Biodefense Science Board, recommended moving forward with a study pre-event, but pending an ethical review. And that's where the commission's work comes into play. This really does set up the fundamental question at the core of the commission's work, which is what research risks, if any, are justifiable to children in this case when the threat of an anthrax attack or a threat of an attack more generally is unknown and perhaps unknowable. Really, this duty to protect children in the event of an attack to the extent possible, while also protecting children from undue research risk. Secretary Sebelius contacted the commission in January 2012, again continuing with this timeline, asking the commission to now conduct this ethical analysis that was left to be done. I think it's important to note here the scope of the charge, that this did bubble up. It was catalyzed by these events surrounding anthrax vaccine, but the commission's charge was broader. The secretary asked the commission to look both at ethical considerations of conducting medical countermeasure research more broadly, and also more specifically, the ethical considerations involved in studying anthrax vaccine in particular. I also want to be clear that there was no protocol in front of the commission. They weren't sitting as an IRB. They weren't sitting as a national level review panel. They were looking at kind of whether any kind of anthrax vaccine trial could ethically move forward at all, and under what conditions it could be conducted ethically, again, if any. So in working on this charge, the Commission sought input from many different areas. It held 4 public meetings around the country, published a request for information in the Federal Register, and as you can see, received several very informative responses. It sought advice from subject matter experts and, of course, engaged the community of the many stakeholders who are interested and very much invested in this area. The result is the Safeguarding Children report by means of illustration visually. This is what it looks like. It's not short, it's not too long, but there's a lot in here, and like I said, it's available for download for those of you who'd like to look at it in more depth. That came out just a little over a year ago in March 2013. I will pause to quickly, have a definitional point. So I appreciate the introduction of framing what medical countermeasures are, because it's certainly not a term I was familiar with before this project really came before us. And the Commission was very specific about the scope of what it was dealing with. And it defined medical countermeasures as FDA-regulated products and interventions used in response to chemical, biological, radiological, and nuclear attacks. Now, I just note this because definitions of medical countermeasures can vary, so I just want us all to be on the same page about what I'm talking about. Some entities define it to include non-FDA regulated products, for example, and also other entities will include responses to naturally occurring disease, and that was not what was on the table for the Commission, given the context in which it received its charge. So the Commission wasn't writing on a blank slate, and I am sure these 2 reports are quite familiar to all of you in the room. There's very strong, well-versed ethical foundations here, especially from the National Commission, really the first in line of the National Bioethics Commissions, of which our current commission is the most recent. We have the Belmont principles that really provide the core for what's going on here: respect for persons, beneficence, and justice. Now, the Bioethics Commission explicitly added a 4th to the mix, and that's democratic deliberation, finding it implicit in the work that the National Commission did, as well as how it did its work, as well as the recommendations it made for research review. In addition, the Commission drew very strongly from the Research Involving Children report, which I think puts the problem into context, several decades ago, that we're looking at the ethical principles that our intention with the obligation to protect the community, as well as the prohibition against using non-consenting persons in research, and how we reconcile those 2, which are especially in tension here. In addition to the ethical foundations, there are certainly regulatory foundations. These come really right from the recommendations that the National Commission made in the Research Involving Children report. And again, I think will look quite familiar to many of you here. The important thing I think that I want to note here is the distinction among levels of risk, which will come into play very quickly as we get into the Commission's analysis, as well as the differentiation of the locus of review. So that there are 3 typical or more typical types of research that are generally reviewed at the local IRB level. This is typically how pediatric research moves forward. Minimal risk research, research with the prospect of direct benefit, and research that presents no more than a minor increase over minimal risk, but is likely to yield generalizable knowledge about participants' condition. Now, there's another section here, Section 407, and I'll note that these are repeated in the FDA regulations as well. I'm going to stick with the Commission's convention of using HHS regulatory citations, but they're also fully in effect in the FDA regulations too. But we have Section 407, which allows for national level review in really exceptional cases with a higher level of risk and no prospect of direct benefit. So all of this leads to what really formed the ethical grounding for the Commission's recommendations, that the government has a duty to both protect children from undue research risk and to be prepared for an attack to the extent possible and protect children in that case as well, that children are ethically and legally unable to consent and therefore require additional protections in research when they are involved. And this really boils down to what the Commission termed the central tenet of pediatric research, that it should be allowed only when it poses minimal risk generally, except when there's a prospect of direct benefit or when there's a likelihood of yielding generalizable knowledge about the participant's condition. And that really is a restatement of what the National Commission said and what's in the regulations currently. So there's another very important ethical distinction along which the Commission framed its analysis, and that's the distinction between pre-event studies and post-event studies, an event being a terror attack like an anthrax attack. And the ethical considerations as well as the practical considerations are quite different in the two scenarios. So in pre-event research, there's been no attack, no one's been exposed, no one is sick. You're dealing with healthy children that have no prospect of direct benefit from the research. And by contrast, in a post-event study, children have been exposed. Children might be sick. Children, in many cases, will receive an untested medical countermeasure already. So you're looking at a very different landscape of research risk. If they're receiving the untested medical countermeasure already, you're really looking at the additional research risk of observational trials. It might be minimal risk, but also because children have been exposed, there's a potential for direct benefit, and there's also perhaps a likelihood of yielding generalizable knowledge. So you're looking at very different places along this ethical and regulatory spectrum. There's an asterisk here that I'd like to just point out, which is the Commission's recognition that when a threat is imminent, the ethical landscape tracks that of post-event research, not pre-event research. And this is really when there's a substantial certainty that the event is about to occur and there's no time for deliberation. You really are in the same type of scenario as if an attack has already happened. So that's just a caveat to have in here as well. So with those distinctions in mind, this is the map of how I'm going to go through the Commission's recommendations and how they shaped their analysis. Really along these distinctions we've already mapped, looking at pre-event and post-event research, and also making the distinction between minimal risk research and more than minimal risk research. So we'll start here, minimal risk pre-event research. Now there are particular challenges that I've already alluded to in pre-event research. There's no attack, no children are sick. I think this is also key in it's a place where many people initially focus when we're talking about medical countermeasure research, that the event has an unknown and again, perhaps unknowable likelihood of occurring. We also, in this case, hope never to have occasion to use the results of the research. We hope that an attack doesn't happen. We hope we never have to use these medical countermeasures. And this leads to the Commission's first recommendation, and that's that generally, pediatric MCM research, pre-event, should pose no more than minimal risk except under extraordinary circumstances. Now, this really is, in a way, just a restatement of the ethical grounding I described earlier, but was the first primary foundational point the Commission wanted to make, which is, if you can do minimal risk research, that's what you should be doing. There are many research designs from which to, start looking at these things, but generally, like in all pediatric research, it should be minimal risk, it should be minimal risk except if there are extraordinary circumstances. Now, recommendation 2 is a more practical take on that, which is, well, how exactly do you do this type of research as minimal risk research? And recommendation 2 focuses on 2 points, which is, 1, do all ethically appropriate prior testing that you can before starting pediatric research. Computer models, animal models, research with the youngest adults. And then, if pediatric research is appropriate, if minimal risk research is possible, to proceed in a manner that the Commission described in quite a bit of detail, but through an age de-escalation process. And I want to pause to just go through a little bit more of what the Commission meant by that age de-escalation, because I think it's really key to how they understood minimal risk medical countermeasure research to be possible. So the idea is really this. If you do all possible research with the youngest adults, 18, 19-year-olds, to characterize the risks of the intervention and the risks of research, to really thoroughly characterize, identify, understand the risks involved, it might be reasonable, it might be possible to make an inference that research with the oldest children also could be minimal risk. And again, when we're talking about research with the oldest children, you're talking about where assent can be particularly meaningful as well. And then doing research with the oldest children to, again, further identify, understand, characterize risk to move down incrementally to the next youngest age group and modify based on what you've learned with the older group and do minimal risk research with that group, and to continue until you can no longer continue anymore. Now, of course, age isn't the only relevant factor here. It's very context dependent. Obviously, developmental categories, how a particular medical countermeasure or agent works. There's a lot to be taken into account here, but the basic premise remains that moving incrementally, learning about risk at each stage, you might be able to proceed in a stepwise, minimal risk manner. Now, applying this to the anthrax vaccine, which again the Secretary asked the Commission to do, the Commission concluded that more steps need to be taken before pre-event research can happen, that we're not in a scenario where minimal risk research is possible at this time, that there's more research that needs to be done with the youngest adults before minimal risk research might be possible. Research on dose sparing, safety, immunogenicity. And in making these observations, the Commission recognized and noted that, for example, mild and moderate adverse events of AVA are no worse than for other vaccines, that there is a potential here for this minimal risk age de-escalation. But again, not looking at a specific protocol, not making any call now except to say that more research with the youngest adults needs to be done to more appropriately characterize the research at this time. So that's this first set. And we move on from that to more than minimal risk pre-event research. And again, it really relies on this key recognition of the Commission, which is, yes, if we can do minimal risk research, if we can do this stepwise process of age de-escalation, we should. But sometimes that's not going to work. That there are times when the inference will not be appropriate. It might be going between age levels, developmental stages, that you just can't infer that the research will occur in the same way, that the intervention will react in the same way with a different age group. It might be that you're looking at that the risks associated with the intervention itself, it could never be characterized as minimal risk. That what you learn from researching with the youngest adults is actually, no, we can't even make that first inference to children. And it also just could be that perhaps there are practical considerations like the amount of time something like this could take. If you know that, an attack isn't imminent, but there might be other considerations involved with how fast you might have to move. So there are the Commission recognized many circumstances in which, you do need to consider this next category of more than minimal risk research before an event occurs. Now, this is when we get back into Section 407, which I had mentioned earlier that we would do. Section 407, again, came out of the work of the National Commission, and their quote on the bottom of this slide, I think, is particularly prescient. Now, they were writing in the midst of polio outbreaks and the development of a vaccine for polio, and used a pending epidemic as their example. They weren't thinking of a bioterror attack, but did note that there might be exceptional situations when considerable dangers to children or to the community at large might be avoided or prevented by exposing to children to research attended by more than minimal risk. And that applies to a pending epidemic, but might well apply to a future terror attack as well. So again, to reorient us, we're talking about this pre-event research where there's no prospect of direct benefit, higher than minimal risk. And under Section 407, these are the regulatory criteria that are there. The Secretary, in addition to national level review, so in consultation with a national level panel, must determine that the research presents a reasonable opportunity to address a serious problem, that it will be conducted in accordance with sound ethical principles, and that provisions are made for parental permission and meaningful child assent. Now, I'm not paraphrasing that much on this slide. This really is the meat of the regulatory language, and as such, I would posit is fairly ambiguous, if only to, well, what exactly are sound ethical principles and how can that be applied in practice? And so that's where the Commission really jumped off from and created this. It's an ethical framework that really has dozens of pages dedicated to it in this report and is summarized in an appendix that I've pasted onto this slide. So know that there's much more than just these bullets that you see here. But what the Commission intended for this to do is to clarify the circumstances in which the research presents a reasonable opportunity to address a serious problem, to specify conditions to determine if research is in accord with sound ethical principles, and to reiterate the importance of informed parental permission and meaningful child assent. So really going into this regulatory language and giving ethical guidance for those who might have to review studies that fall under this regulatory category in the future. So here's the excerpt of where I want to focus within this framework, and that's on the sound ethical principles piece. And you'll see a lot here, and this is one of the places where I certainly want to posit there's a lot here that's applicable outside of the pediatric medical countermeasure context, and perhaps also applicable outside of the Section 407 context as well. The Commission expanded on criteria, for example, for ethical research design, for subject selection, for community engagement, for transparency and accountability. But where I really am going to focus now on are the two that you see boxed here, A and C, the ethical threshold of acceptable risk, and also the post-trial requirements, which I think actually have very different stories perhaps of more general applicability. And I should say the Commission recognized when it put this framework forward that it might have applicability outside of the medical countermeasure context, even though that's the context in which the Commission developed it. So the first of these criteria, this threshold of acceptable risk, after saying all of this about more general applicability, I'm going to say this is probably the one criteria that is quite specific to medical countermeasure research. And Section 407 does not state a threshold of acceptable risk. It's currently obviously an issue discussed in the literature. What really is the threshold for the amount of risk you can present to a child in these exceptional circumstances. But that aside, given the specific context of medical countermeasure research, the Commission said there is a clear limit, and that's minor increase over minimal. It's a threshold that's familiar in the regulations. It's applied in Section 406 as well. And the Commission really applied it here based on, again, the circumstances of medical countermeasure research, that we're talking about a situation in which ever needing the research, ever needing the fruits of the research, applying the research, you don't know that the threat of an attack is unknown or unknowable. I know I keep saying that, but that really is the crux. To, assume this unknown, assume the unknowable, but still figure out how you might be able to ethically move ahead. And where the Commission really brought all of that together is in this threshold of risk and saying, okay, we're not ever likely going to know whether an attack is going to occur. But that said, because of that circumstance, the threshold of risk should be limited to what's listed here, this minor increase over minimal, which still would pose no substantial risk to the health or well-being of the children involved. So that's that first criteria that we're focusing in on. The second is the opposite story, and I think is something that might be applied more widely, and that's this set of post-trial requirements. So the first listed here is this idea of having a distribution protocol tested or assured in order to move forward. And the idea behind that is that this research in which children would be participating at no prospect of direct benefit to themselves is really for the benefit of future children. If the research proves successful, it's so that there are tested medical countermeasures available for children in the future in the event of an attack. Now, for that benefit to accrue, those medical countermeasures would need to be available to children if they were needed. And that's where this ethical requirement comes from, that you need to have at least a plan in place to assure that the future benefit, if it exists, will accrue to the children for whom this research is being conducted. And so, in other contexts, in other 407 contexts, it might not be a distribution issue, it might be a translational issue, it might be something else, but this idea of planning for the benefit for future children, I think, is an important one, especially as part of the justification for 407 research more broadly. The Commission also said that there must be a plan in place and there must be assured compensation for research-related injury in this context. Now, this is not a new issue to national bioethics commissions. It's not a new issue for this bioethics commission. They did more in-depth recommendation on compensation for research-related injury in an earlier report. But they also felt it was very important to raise it again here. That again, we're talking about children. We're talking about children who have no prospect of direct benefit from the research, that the rationale for compensation is particularly acute here, and it should be in place as an ethical condition for moving forward. Now, the Commission didn't say that there needs to be a standard mechanism in place for compensation, that there needs to be a national mechanism. It could be at several different levels, whether it's government established, particular to the funder or to the research site, but it must be in place to ethically move forward with pre-event research. And that leads to the next two recommendations. So first is the set of preconditions, in a sense, to 407 review, that minimal risk research must be impossible, and the research should pose no more than a minor increase over minimal risk. And you'll see on the slides, and I've been glossing past this a little, but that the Commission did make an attempt to specify a little bit more about what minimal risk and minor increase over minimal risk mean. Again, I realize the group I'm speaking to doesn't need to hear that these are not necessarily clearly defined, and the minor increase, less so than minimal risk. But the Commission tried to give a little bit more substance to it as going through, recognizing that these aren't immediately clear terms. And some of the maybe most fruitful discussions that the Commission had with experts in the field who came to these public meetings was about how you think about risk and levels of risk. And I think really the Commission benefited from those conversations and input. And then we have recommendation 4, which is that should the research proceed to national level review under 407, that the Commission's framework should be applied. And that's the framework that I showed you in outline form that we talked through a little bit, and really is some of the more ethically and analytically heavy work here, and asking that it be, or recommending that it be applied should this situation come into play. Now we have the application to the anthrax vaccine, again, the specific request of the Secretary. The Commission concluded that, again, having not yet being in the position to know if minimal risk ABA research is impossible, given what we talked about just a little while ago, if it's ultimately determined that it is impossible, the research should proceed to national level review, the risk should be capped, and the ethical framework should be applied. So now moving on to, again, very ethically distinct scenario of post-event research. I'm not going to talk about this as in-depth as pre-event. It involves, for example, the FDA regulations for emergency use authorizations, investigation of new drug applications. As a recovering attorney, I'm happy to talk about the intricacies of regulations, but I'm going to assume that might not be nearly as much of interest as the conversation I hope we can fit into at the end of this session. So, again, recall here, children have been exposed, most likely receive, or at least in many cases already receiving an untested medical countermeasure. I think what's important here is that it simply really doesn't matter what you've done pre-event, because there's an ethical imperative to do research when the attack happens in either case. If you've done research pre-event, you've done a very small, very targeted study. Now you have that medical countermeasure being given to children in a much broader way. There's an obligation to learn as much as possible in that case, to get more adverse event information, for example, to do the surveillance studies, to learn as much as you can now that you're giving it to children on this scale. If you haven't done any pre-event research, then it's also ethically imperative to do this research post-event, to learn what you can, because again, the untested intervention is being given to children in any event. So we have in the post-event case also particular scientific and logistical challenges. So you have this different ethical landscape. You also have a very different practical landscape. You're looking at very stressful circumstances, to put it mildly, I realize. But it's a very different scenario. You have parents and children in different places, and there's a lot going on to just get whatever research part of this up and running. There's also the scientific challenges. You have many variables, confounding variables, interventions that are being given together. Recall that anthrax vaccine would be given in addition to antibiotics. You don't have this small controlled trial. You're really relying on a larger observational trial. And that leads to the Commission's recommendations around post-event research. So the first is that post-event research should happen, and it should be planned in advance to the extent that's possible. There are mechanisms, again, in the FDA regs that allow a lot of this to be planned before it occurs, and that there are a lot of kind of ethical pieces that are important to have in play, and that's what this list is at the bottom. In particular, the Commission pointed to these things that IRBs should ensure in doing any kind of review of post-event research, because again, recall that this is research that likely will be approvable on the local level, that there are adequate processes, of course, for permission and assent, that there's a scientifically sound design, participants have access to the best available care, that adequate plans are in place to treat or compensate research-related injuries. Again, you can see that coming up again here, and that there are provisions made to engage communities, to really to speak with stakeholders, speak with the communities, to be clear about what they need from the research and also why the research is being conducted. And here we have some of those lovely acronyms I referenced earlier, but this is regulatory, but it's got a broader context, which is basically the conclusion that if post-event research takes place with children on untested medical countermeasures, so where there's no data available for these interventions with children, when the MCMs are given to children post-event and when they're researched post-event, full research protection should be in place. Again, this comes out of these regulatory mechanisms, but it was, I think, important to the Commission that if you're using untested medical countermeasures, full research protections, full review, full consent processes with permission and assent are in place given the state of the science. So applying that to the anthrax vaccine, there are plans in place. Right now, there's a treatment IND held by FDA and CDC. So basically, a way in which to distribute the medical countermeasure, the anthrax vaccine, broadly to children if an attack were to occur. And also, the FDA and CDC are working on a nested research protocol. So again, it's under a different type of IND, but given that the MCM will be distributed broadly, there are plans in place to do a smaller study within that to observe what's going on as children are using it. Both mechanisms, again, require full research protections, IRB approval among them. So these are the few lines at the end of the Commission's report that I think are important to conclude with here as well. Which is that pediatric medical countermeasure research brings into sharp focus the fact that the health and security of children are paramount. Such research warrants an ongoing national conversation in order to ensure the highest standards of protection for children that reflect an unwavering commitment to safeguard all children from unacceptable risks in research and through research that promotes their health and well-being. So I want to thank you for being here for this breakout and also for what I hope is a little bit of discussion ahead to really continue this national conversation that the Commission's report is part of and that we hope will be ongoing as these issues continue to play out. So thank you. These are the many ways in which to stay in touch with us. Please do. And let me also just mention that all of our meetings, our webcasts, we actually have one coming up in August in DC and another in November in Salt Lake City. So if you're interested in what the Commission's doing, please do stay in touch. Please do check out those meetings and please do let us know what you think about the work. So with that, I'll pause. Thank you, and hopefully have a little bit of time for questions and discussion and to hear from all of you.
Terrific presentation and I think a really difficult problem. I had a couple of questions as others of you are thinking about your thoughts and questions you might have. They're related. So I was wondering, to what degree did the Commission discuss the problem of transparency? And it seems to me it affects this issue in a number of different places. So the first is for the pre-event research. A lot of the information that's essential, for example, for the ethical review, but then let's say it's approved for the deliberation of communities or for individual parents, is not necessarily available in terms of, what is the likelihood of risk. A lot of these discussions involve national security. It's very difficult to have full information in the way in which we would if you were looking at an infectious disease outbreak, for example, where we have lots of surveillance data. It's wide open. We have a better sense of the risk. So that's the first place where I think transparency seems really important, or lack thereof. The second is deliberation, right? You mentioned that deliberation was one of the main principles, but without transparency, how can we have a very open dialogue, either as a community or a nation, about where our funding priorities ought to go for pediatric research? So, you know, how could we have an open discussion about the level of risk of an anthrax attack and whether we want to put federal dollars there, or whether we want to put federal dollars towards, you know, pre-5 routine vaccinations in, you know, impoverished families? You know, how can we really genuinely have that public deliberation without transparency?
So thank you, and thank you for that question. I think transparency and accountability those were core to the Commission's analysis, and I think it comes out in at least two ways. So the first, really in direct answer to the first question you posed, which is, well, a lot of this information perhaps is going to be classified, you know, either not known or not knowable to the public, or even maybe folks who are involved in the decision making. And the Commission addressed that as, I believe it's part of the seriousness prong of the ethical framework, saying that, you know, recognizing that, yes, a lot of this information might be confidential, but saying, one, these determinations need to be based on best evidence, that there are models that are used within Homeland, within other agencies in the government, that these can't be based on fear alone, that the assessments need to be based on the best evidence available. Also, that to the extent that they need to be based on the best evidence. Also, that the Secretary is accountable to the public. And while there might be confidential details that can't be shared, there needs to be some sort of public discussion about the reasons to determine that there's a threat. Now, that doesn't mean giving the whole threat analysis, but describing to the public what can be described about the threat, and also, perhaps, if that's not sufficient, to make disclosures to authorized representatives of the public, even, you know, Congress, other representatives in government. So recognizing that, yes, there's a lot of concerns around that threat determination, but saying what the Commission can say, which is, one, base it on the best available evidence, and two, you can't hide that rationale behind confidentiality, that there needs to be, or behind classified information, that there needs to be public accounting of that rationale, be it to the full public or be it to representatives of the public. The other piece of transparency and accountability here came into play under the sound ethical principles piece, which is more to this idea of deliberation, which is that Section 407 allows for public review, public comment, and really focusing in on that as being critical to this. Also, the Commission pointed, interestingly, to the composition of the national level review panel, that there's a need to have a varied representation there, to have people with different interests, different perspectives, and to have that be part of the transparency and accountability as well. So this theme runs throughout the report. As to the bigger question of research priorities, this might be an unsatisfying answer, so realize that I recognize it as I say it, which is, you know, that wasn't the decision the Commission was making here. That this wasn't a report about the priority of medical countermeasure research within a broader public portfolio, that's a decision that lays in other parts of the government. This is really a question of, can this move forward ethically, and if so, how might it move forward ethically? So, you know, hopefully these pieces of the conversation that the Commission really focused on, being transparent to the public, being available to the public, especially given the particular nature of something like pre-event research, hopefully that being out there will foster this broader dialogue, but that really wasn't a broader question. That was part of the Commission's charge in this report.
Can I just ask the general question? Given the time that's elapsed since the Commission released its recommendations, how have they been received, and what have been the subsequent developments in the area, particularly around, say, research in young adults?
Sure. So in the how has it been received category, I think generally it's been received well by the various audiences we've been speaking with. I think this is an issue on which there are lots of views all along the spectrum, which I say only to say that people have very strong views on, you should never do this, and why aren't we moving ahead with this full speed ahead? Why are we even having this conversation? You know, those are obviously caricatures, but, you know, really do represent a spectrum. And I think, you know, the Commission really did try and weave a very careful and thoughtful middle ground, and generally the stakeholders that we've talked to about this and that we've had conversations with, I think, see it that way. See it as not a full stop, not a push ahead, but a carefully considered way to move forward if we're going to move forward. So I think we've had a very a positive reception in that way. I don't know what's going on with young adult research. I think, you know, that sits within other parts of the government, and nothing that I've seen kind of published or anything like that. So I confess to not knowing what's going on there. I do recently the AAP came out with a paper more on clinical presentations of anthrax with children and how to treat it, but there is mention of public health preparedness and the Commission's age de-escalation recommendations and the potential for moving forward with this type of age de-escalation research. So there's at least discussion in that community as well of moving forward in that way, and that's something that I've seen recently too. But this is, you know, still obviously a very open issue, and we continue to watch with interest what's going on.
So I had a question about minimal versus a greater or minor increase over minimal risk for vaccines. And every biologic study that I've ever seen, it's always been greater than minimal risk. So what scenario would you see using an anthrax vaccine that would be minimal risk?
So the Commission's reasoning was this. So first, that we don't know. We don't know whether it can move forward yet as minimal risk, but the possibility still exists. And that was based on really a review of currently the reported mild, moderate adverse events, that they really are quite similar to other routinely administered vaccines. And looking at that, plus the additional research risks, it might be possible to do minimal risk research, but we don't know enough yet about this research with the youngest adults to be able to even make that inference, but left open the possibility that it might be, and that's based on looking at, for example, adverse event data and comparing it to other routinely administered vaccines.
It would be pretty remote. I mean, vaccines have known side effects, for instance, like the pneumococcal vaccine being developed in India. It's almost a known entity, yet it's considered greater than minimal risk with the potential for benefit because these children are being immunized against pneumococcus. So I find it hard to see how a vaccine study, I can see other measures, but a vaccine study would be minimal risk.
No, thank you. And I'm obviously not a vaccine researcher, but I think it's important that the Commission really did look at this not as children as 0 to 17, where I think it's particularly hard to make any claim about something being minimal risk, but looking at children as very discrete stepwise groups where it might be possible. But again, that's medical countermeasures research more broadly, and also the Commission said that it didn't have the data necessary yet to know whether ABA trials can move forward. So thank you for the comment. And I'd also, you know, again, invite comments from any of you on thoughts about broader applicability of this. I know it's a big ask on a Saturday morning, but please feel free, if you have comments as opposed to questions, to share those as well in the last few minutes we have. Yeah.
To what degree was this discussion international at the Commission? Did you reach out? I mean, WHO has been worried about this, other countries have been worried about this. Is there attempt to coordinate efforts? These types of events don't always respect boundaries.
So that's a question that actually I'd be happy to chat afterwards, because I actually want to look at the speaker lists, which I'm not going to do while I stand up here. But what I can say more broadly is that the Commission has engaged internationally on all of its projects moving forward and is very keyed into hearing what's going on internationally. I think, you know, I don't have it off the top of my head, but I would be really surprised if we didn't receive international comments in response to Public Federal Register, because typically we do. We hear from, you know, a lot of our, you know, of other bioethics commissions, of other people involved internationally on a regular basis through that mechanism, also by inviting international experts to come speak with the Commission. So that said, it's not on the tip of my tongue of the exact examples in this case, but other than to say that the Commission's been very involved internationally with bringing international experts, hearing international perspectives in all of its projects throughout its tenure. All right. You look about to do it.
So then can I ask, so like on slide 17, when you're talking about minimal, a small increase over minimal risk,
Right.
the minimal risk category is generally characterized in terms of like activities of daily living for children. And risks could be considered both in degrees of frequency as well as severity. And certainly there's lots of things in children's lives that are high risk but infrequent. So playing football could involve a spinal cord injury, which would have a severe consequence for a child, but is relatively infrequent. But the way the language on slide 17, the emphasis was predominantly on severity and not frequency. And your later comments are about kind of, well, in comparison to other vaccines, which may actually have fairly severe consequences, but they're infrequent. And so when the commission thinks about minimal risk, how does it balance these severe consequences that are infrequent in children's everyday lives?
Sure, thank you.
And is it really just capping it at the severity, or is it willing to accept severe consequences as long as they're phenomenally infrequent?
So the Commission certainly recognized the many things that go into the concept of risk, things like frequency, severity, that it's not just a singular concept, that there are many inputs to that. So there is certainly discussion of that, certainly a look at that. I think this comparison to other routine vaccines really comes out of the part of the definition of minimal risk that focuses on routine medical examinations. That, if a vaccine is part of routine care, then perhaps within that definition of minimal risk, that's where that comparison comes into play. So the Commission certainly looked at this as broader than something than just severity, just frequency. It has a much more fleshed out view of risk in the report itself, but that comparison to adverse events comes along that rate of minimal risk. So with that, I think I might have the power to give you an extra 3 minutes of break. And again, thank you very much. I'll be around for at least a few minutes if anyone wants to chat more. But thank you again for being part of this. And please don't hesitate to be in touch with me or the Commission should you have further thoughts on this.
Welcome back. We are ready for our final session of the morning. And to do that, I'm going to welcome back Alex Capron, who we introduced yesterday, but I will reintroduce. Alex is a professor and the Scott Vice Chair in Healthcare Law Policy and Ethics at the Gould School of Law at the University of Southern California, where he also teaches at the medical school. And Dr. Capron has been working in this arena for many decades and has had involvement on at least 2 presidential commissions on bioethics, as well as many other sort of national responsibilities. So he is going to talk about young patients and age-old issues.
Thank you, Doug, and let me express my thanks to you and to Ben Wilfond for inviting me, but not for putting me on today's program in a position parallel to Eva Moses Kaur. What an extraordinary life story she shared with us. I do hope that you all take to heart her message of forgiveness and begin to apply it immediately, such as to my presentation. For all its differences, my talk does have in common at least 2 things with Eva Kaur's presentation. First, my remarks are not confined to issues of pediatric research, but rather deal more broadly with research with human beings, and then propose the pediatric aspect as an amplifier of the issues that I will broadly identify. Second, I will be inviting you to think again about the roots of the present regime for ethical review of human subjects research. I suppose that much of what I have to say will be familiar to you and may also seem like ancient history. Yesterday, Norm Faust suggested that what has been derived from the historical experiences I will mention is now just symbols whose original meaning has been lost, so that they now serve merely to identify a certain tribal allegiance and are mere shibboleths. I too am concerned that some of our ethical commitments are now honored verbally but not in practice. But I don't share Norm's conclusion that they are nonsense on stilts and cannot be taken seriously. Instead, I will echo Eva Kaur's caution that doctors must, as she said, walk a very narrow line when they move into research. I've already given you some idea of why I gave this talk the title, Young Patients, Age-Old Problems. But now let me delve a little deeper into the age-old problems. I believe we are now entering a time of uncertainty regarding research ethics, in which answers that we've provided in the past to some basic questions are being brought up for re-examination. It was nearly 45 years ago when Jay Katz and I were working together on what became the first casebook in this field, Experimentation with Human Beings, which was published then in 1972, that we composed the following question: when may a society, actively or by acquiescence, expose some of its members to harm in order to seek benefits for them, for others, or for society as a whole? Today, a number of leading bioethicists have suggested that we give a new answer to this question of when. The immediate provocation is an embrace of the drive to improve medical practice by studying its effects on a continuous basis and feeding the findings back into practice immediately. That is, the learning health system that Steve Joffe described yesterday. Further impetus for this reexamination comes from criticism of the approach that OHRP adopted in dealing with support, as Nancy Cass described, as well as broader doubts about the worth of IRB review generally, and more fundamentally about the importance of informed consent. I plan to explore where we stand on the when question, not narrowly in light of the current debate provoked by support, but instead as a product of the historical evolution of the prevailing ethical paradigm for research with human beings, which seems at times to be the result of a pendulum swinging from one pole to another. Let me suggest that we can understand this history by looking at 7 times when the ethical and then the legal paradigm shifted. Let me lay these out quickly, and then I'll elaborate on each one. So these 7 paradigm shifts begin with the period before 1900, where we had heroic physicians and the beginnings of scientific medicine. And then around 1900, there were some legal remedies that were created by the state and that were created by physicians. Obviously, in the period from 1932 to 1945, we moved into some very dark days, and then the reaction to that came at Nuremberg. In the period between 1947 and 1964, there was a retreat, I believe, from the Nuremberg ethics, and that was followed by another swing of the pendulum in the opposite direction in the following 12 years, with the recognition of those problems and further efforts at reform. The present era is one in which we have added complexity to the whole process of research ethics review and a culture of compliance. And that's led, during the past 10 years, to calls for the reassessment and a new ethics that I want to address. So let's begin then looking at the way this could be described. And I'm going to put this up quickly and come back to it. And in doing this, I want to make clear that what I have here is something that has to be understood as kind of a metaphorical representation. It's not an empirical representation, but it is intended to indicate the rising and falling level of ethics over the period that I've just described. And then we have a question mark where we're going today. So let's look at the first paradigm. The core question for 19th century physicians who aspired to be scientists was how they could become researchers and still remain true to the Hippocratic injunction, primum non nocere. Perhaps this is the first of the shibboleths that Norm attacked yesterday, though I believe it is possible to do both. In any case, in his great work published in 1865, An Introduction to the Study of Experimental Medicine, the renowned French physician-scientist Claude Bernard echoed the earlier injunctions of Immanuel Kant and Moses Maimonides in counseling his fellow physicians not to treat their patients solely as means of advancing knowledge. He did not suggest, however, that the subject's informed consent be obtained. Yet he did argue that physicians must always keep the welfare of patients foremost when conducting research. Around the turn of the 20th century, several events brought that medically focused paradigm where it was doctors taking the heroic step, taking the risks on behalf of patients into their own hands. And that era came something to an end. And it began with the research carried out by Albert Neisser, the famous Prussian physician who had discovered the bacterium responsible for gonorrhea, when he inoculated in 1890 15 women and girls, one as young as 10 years old, with a cell-free serum from patients with syphilis. His hope and intention was that they would be vaccinated against the disease. None of those to whom he put these injections knew what the experiment was about. Many became infected with the disease, which at the time did not have a cure. The public outrage, once these injections became known, his work became known, led to both moral censure of him and a criminal fine, and 10 years later, in 1900, to a directive issued by the Prussian Minister for Religious, Educational, and Medical Affairs, an interesting combination, requiring that the possible negative consequences of each experiment be examined and that unambiguous consent be obtained from all subjects. Notably, this meant that no research could be conducted in orphanages. Also in 1900, Dr. Walter Reed was leading a team of U.S. Army physicians conducting research on yellow fever in Cuba, which had recently been taken from Spain in the Spanish-American War. A number of Reed's colleagues allowed themselves to be bitten by mosquitoes. When two of these Army doctors became infected, and one of them, Dr. Jesse Lazier, died, the investigators decided to switch to enlisted men and Spanish laborers. Reed was very aware of the charge leveled by William Osler that the yellow fever experiments done several years before by Santarelli on 5 immigrants in Uruguay were criminal because the subject's consent had not been obtained. Therefore, Reed developed a written consent form. It identified study risks, principally that the participants may develop yellow fever, but also placed those risks within the local context, namely that they were very likely to get yellow fever anyway. The loss of life during the Spanish-American War was principally due to disease, both among the Americans and the Cubans and the Spaniards. The contract also promised that if they did get infected, they would benefit from the care that the researchers could provide, which would be superior to any medical treatment they would otherwise obtain, particularly the Spanish laborers. The form promised the laborers a payment of $500 in gold, which would be doubled for those who developed yellow fever. And this was, of course, a very large sum at that time. Thus, as of 1900, with this second swing of the pendulum, the answer to the when question was that research is acceptable when legal requirements, either set forth by the government in legislation and regulations or in contracts that imposed obligations on the researchers and required the subject's consent, had been met, and in particular, when competent persons knowingly agree to participate. The frailty of this paradigm became apparent in the horrors of the Nazi concentration camps that were so vividly described yesterday. For the Nazi researchers and their Japanese counterparts studying diseases, seeking medical information for use in their war efforts, and at least in the notorious Japanese Unit 731 in Pingfang, China, also testing chemical and biological weapons. The answer to the when question was when it serves the interests of the state. What we most remember from those dark days, however, was the reaction, in ethical terms, in the articulation of what we know now as the Nuremberg Code. That was promulgated by the military tribunal as it passed judgment on the Nazi doctors in 1947. The 10 principles stated by the judges to explain when medical research with human beings is ethical began, of course, with one of those statements, the voluntary consent of the human subject is absolutely essential, that Norman Foss told us yesterday can't be right because we couldn't have biobank research or research on patients receiving emergency care or research on children if that statement were true. In some ways, the Nuremberg Code marked a high point in the development of research ethics. But as later became apparent, even as the American judges were pronouncing it, American researchers were carrying out studies that fell far short of the Code's commandments, such as the wartime research in U.S. prisons, and most particularly the Tuskegee syphilis study carried out by the Public Health Service beginning prior to the war in 1932 and carried out for 40 years until it was splashed on the front pages of newspapers by an investigative journalist. As with the second paradigm, the Nuremberg Code had neither a pervasive nor a truly lasting effect on the conduct of research for several reasons. First, the post-war period produced triumphalism in medical research, with the public and its representatives in awe of the new antibiotics, vaccines, and medical technologies that seemed on the brink of conquering disease, just as Imperial Japan and the Third Reich had been conquered. During this period, besides such things as STD experiments, not just in Tuskegee but Guatemala, boldly carried out by the government's physician scientists, even if not openly reported at that time, and the rapid growth of investigator-initiated studies, the archetype of the new science was the development of the randomized clinical trial, beginning with the trial of streptomycin for pulmonary tuberculosis in Britain, conducted, of course, without the knowledge of the patients enrolled. During this period, the when question was answered not only by saying that some patients could become the means of developing knowledge when physicians felt that a research goal was important and could feasibly be achieved, but also when national needs dictated. Many activities, not just the testing of atomic weapons, but also medical research on civilians, such as the ionizing radiation and LSD and related experiments, rested on this Cold War rationale. Equally significant, the World Medical Association promulgated the Declaration of Helsinki in 1964. This document is usually described as establishing high ethical standards, indeed standards so daunting that they have to be torn down as mere shibboleths by the critics. While I certainly share the view of many that the Declaration has never been a completely coherent document or one that can easily be followed, given its inconsistencies, what is most important, in my view, to remember about the Declaration of Helsinki is that it amounted to a major retreat from the Nuremberg Code. Organized medicine, particularly the German Medical Association, seeking to rid itself of the Nazi taint, wanted no part of a set of principles developed by judges in the concentration camp context. Instead, with the Declaration, organized medicine took back authority to set ethical principles for human research. The drafters of Helsinki drew a sharp distinction between research combined with medical care and research on normal volunteers. And as to the former, they continued the existing paternalistic view that physicians should tell patients enrolled in research only as much as they, the physicians, thought it would be good for them to know. That era of the retreat from ethics began to come to an end in 1966 with a shift from the fourth to the fifth paradigm, which continued to develop over the next dozen years. And there were two events in 1966 that really marked that as a watershed year. The second one, actually, chronologically, but the one which has always gotten the most attention, is Henry Beecher's publication in the New England Journal of his article, Ethics in Clinical Research, in which he recounted in detail 22 experiments which he regarded as unethical, principally because of excessive risk or because there was not consent from the subjects or knowledge of the subjects of the fact that they were in a research experiment, or both. And these 22 were drawn from what he said was a longer list of 50 examples that had been provided to the editors. Earlier in the year, Surgeon General Stewart required that public health service researchers, physicians carrying out research who were employed in the public health service, would have to have prior review. And that prior review was initially from colleagues. It was peer review, but within a short period of time, non-scientists were added to the process. And then, of course, eventually that was expanded outside the walls of the public health service to public health service funded research. 6 years later, the Tuskegee syphilis study was revealed, and a process began in the United States Congress with looking at that study and other examples from the 1960s onward of research that raised ethical questions. The Senate hearings were held very prominently the following year in '73, and in 1974, Congress passed the National Research Act, which incorporated the provisions which had once been intended as a separate freestanding bill, creating the National Commission for the Protection of Human Subjects of Biomedical and Behavioral Research. And besides creating that commission, the law required every institution receiving federal support to set up what it called an institutional review board. That's the origin of the term IRB, and required that there be consent of subjects. As a result of that commission's work, a number of reports on specific areas of research were published, and that culminated in their response to the charge that they were given to state the ethical principles underlying ethical research. And that was the Belmont Report, named after the conference center in Maryland, Belmont House, called Ethical Principles and Guidelines for the Protection of Human Subjects of Research. We all know the 3 principles articulated there and later expanded to 4 by Tom Beecham, one of the staff members with principal responsibility for writing that report. The principles of beneficence, and now beneficence and non-maleficence, respect for persons and justice, and an explanation of the distinction between research and the routine practice of medicine, something that the Commission was also asked by the Congress to explain. When we think about the need for the Commission to address that, we should keep in mind that the federal government regulates research that it sponsors, but it doesn't regulate the practice of medicine. And so that distinction, which some people have suggested has a philosophical basis, doesn't have a philosophical basis. It's a jurisdictional basis. Congress, in the process of writing the statute, and then the Commission in responding to it, was well aware that organized medicine in the United States did not want to be regulated by the federal government. This is an era where doctors were very sensitive to that. It was the first decade of Medicare and Medicaid, and there was already a lot of concern about the extent to which payment regulations would amount to ways of regulating medical practice, and this was going to be a sharp line. The ordinary practice, the routine practice of medicine was on one side, and only things which qualified as research. So we had to have the now famous definition of research to separate those two. Now, many people would probably say that that paradigm continues through the present. After all, do we still not turn toward Belmont and bow when we recite our bioethical prayers on a daily basis? Yet I think that in 1978, there was actually another change. This involved the creation of the President's Commission for the Study of Ethical Problems in Medicine and Biomedical Behavioral Research, the successor to the National Commission. It also involved thereafter the first response from the Department of Health and Human Services to the report of the National Commission, which was the creation of the basically the present format of the regulations in 45 CFR Part 46. And in 1981, the President's Commission, which actually didn't start in 78, didn't start until 1980, issued the first of the 2 biennial reports it was supposed to write on research regulation. And in that report, it urged that all government-supported research be governed by a single set of rules. There were then we identified, I was the director of that commission, so I say we identified 20-some different departments and agencies that had rules, recognized that most of the differences, as far as could be told, between the rules were wording differences. They were not in-principle differences. But if you're a conscientious IRB and research comes before you and you're used to looking at HHS research, and this is going to the Department of Defense or the Department of Education or the Department of Justice and on and on and on, you wondered, are we doing the right thing in reviewing it? And if it wasn't clear where a research proposal might end up getting its funding, the same set of questions arose. And so we said, we should have a single set of rules. I guess it's one of those be careful what you wish for moments. And indeed, a couple of years later, there were additional protections for human subjects were issued as an additional part of the federal rules, and the Federal Advisory Committee on Science and Technology created an interagency human subjects coordinating committee, which was chaired by Charlie McCarthy, the director of OPRR, the predecessor of OHRP. And it engaged in a process which took it a mere 8 years, 10 years from the recommendation from the commission, but 8 years from the establishment of that committee to come up with the common rule. And one of the concerns that we've all had ever since then is, could we ever get the common rule changed to reflect experience and new thinking or whatever? And the answer from the federal government has been, are you crazy? It took us 8 years to put this together. How could we possibly do that again? And as we all know, it is now, I guess, 3 years since it looked as though something 20 years after the fact might actually be happening with the advance notice of the proposed rulemaking. And it's taken 3 years, and we haven't yet seen a notice of proposed rulemaking, so that skepticism about the ability to revise the common rule may be well taken. And then the Advisory Committee on Human Radiation issued its report, which led to the creation in 1995 of the National Bioethics Advisory Commission. And again, that commission argued that it was important to have independence between the body overseeing research and the funders of research. And on that commission's recommendation, OPRR became OHRP and was moved to the Office of the Assistant Secretary for Health. Again, not entirely clear how that is playing out. Now, what that I think has meant is that we have an increasingly complex and compliance-oriented world of research oversight. And that could go on forever, but there has been, as I've suggested, a current call for re-examination. And so that's where we are now. That existing sixth paradigm is now potentially up for revision, and not just through the notice of proposed rulemaking. So this brings us really to the heart of what I want to do today, which is to ask, for example, what constitutes research? That's up for grabs. Obviously, when the OHRP sanctioned the Michigan Hospital Association and all of its participating hospitals for the Keystone Intensive Care Unit study, using the checklist approach to avoid central line infections, they got huge pushback. And the question, how can you call what amounts to a quality improvement project research? Not a resolved issue. OHRP was beaten up enough that they backed off as to that. But if you're working in this area, or you have people at your institution that are working in this area, you know how nervous people are that sanctions could still be issued. Further, the question that Nancy explored with you yesterday, what creates risk? Does randomization create risk? And what consent is needed? Is it consent to the underlying interventions, or are those part of a treatment relationship? And all the consent has to address is any added risks, if there are any, from being in a research comparison of existing medical practices. And then the general issue that Steve raised yesterday about continuous learning by studying the outcomes of ordinary treatment, the learning health systems. So those who today want to break and reform the prevailing paradigm would answer the question with which we began: when may a society, actively or by acquiescence, expose some of its members to harm in order to seek benefit for them, for others, or for society as a whole, by saying either what harm, there isn't any, or by saying when those who would do this are acting to prevent avoidable harm, or are acting for the benefit of all in a way that would not otherwise be achievable. And they would point especially to studies conducted within learning health systems as the archetype of the sort of research that is not only needed and justified, but that requires a change in the norms of research ethics and especially ethical review. And this then has led to an even more negative critique, and I am going to be quoting here from Simon Whitney, who had a provocative article that many of you from the pediatric field probably noticed a couple of years ago in Pediatrics, called the Pythons Embrace Clinical Research Regulation by IRBs, in which he claims that IRBs add nothing but expense. They save no lives, unlike researchers who do, that informed consent is expensive and pointless, especially in studies of existing practice in which interventions are routinely provided without specific consent in everyday practice. In other words, reasoning backwards from the failure to get consent in medical care to saying, why would we then burden researchers with this obligation? And consent, if required, will bias trial entry, and he points to the fact that in the support study, a larger number of African American parents declined to participate, meaning that the results of support are more applicable to white infants than to blacks, despite the latter group's higher mortality, and that this therefore violates the ethical principle of justice. And a more cutting criticism from Carol Heimer, which says the biggest ethical lapse in American regulation of human subjects is the death and suffering that has resulted from slowing the pace and altering the focus of research and squandering of research funds. So I want to take that as the challenge. That's the call for a reexamination. But I want to say that a reexamination could operate from exactly the opposite pole. And I want to take us back, if you would please, to the thinking of someone who was also, like Eva Kor, deeply affected by the Second World War and deeply reflective about its lessons, especially about the abuse of power and authority. What I'm going to draw from is something that many of you may be familiar with, which he spoke about in most detail in this contribution to a 1967 conference held by the American Academy of Arts and Sciences, which appeared as an issue of the journal Daedalus in 1968. And that is the article by Hans Jonas on philosophical reflections on experimenting with human subjects. Now, Jonas was not against biomedical research. Indeed, he accepted the usual ethical justifications for conducting research, that it's all right when risks are minimized, when there's informed consent, and a worthy objective. These were the views that came to be accepted post-1966. His further qualification, however, was unusual. He said that research was justified when all of the participants had identification with the goal of the research. And therefore, the best subjects were researchers themselves, either the researchers conducting that research or those who were colleagues, because they would be most identified with this goal of advancing knowledge. He recognized, however, that at some point in research on illnesses, patients have to be subjects because you have to be seeing what the effect of the intervention is on patients. And so he thought they should only be used for studies of their own disease because they would also have identification with the objective there. And he was reciting examples of and attacking the use of subjects who were simply hospitalized patients or otherwise under the control of physicians and used for research not connected, even when they had formally given consent. But in describing the obligations of patients and physicians towards research, he set some very stringent limits based on the following rationale. The destination of research, he wrote, is essentially melioristic. It does not serve the preservation of the existing good from which I profit myself and to which I am obligated. Unless the present state is intolerable, the melioristic goal is in a sense gratuitous, and this not only from the vantage point of the present. Our descendants have a right to be left an unplundered planet. They do not have a right to new miracle cures. And then he went further in this famous passage. If some of the implications of my reasonings are felt to work out toward a slower rate of progress, this should not cause too great dismay. Let us not forget that progress is an optional goal, not an unconditional commitment, and that its tempo in particular, compulsive as it may become, has nothing sacred about it. Let us also remember that a slower progress in the conquest of disease would not threaten society, grievous as it is to those who have to deplore that their particular disease has not yet conquered. But that society would indeed be threatened by the erosion of those moral values whose loss, possibly caused by too ruthless a pursuit of scientific progress, would make its most dazzling triumphs not worth having. Now, given the amount of research that was going on, as you recall, not yet revealed or widely known in the 1950s and 60s, his comments seem almost prophetic in describing the harm that was done then and that was only later acknowledged and for which apologies had to be issued by the government to the victims of the Tuskegee experiments, to the victims of the radiation experiments, to the victims of the mind drug experiments, and so forth. So the question is Jonas's view totally radical, and is it irrelevant? As researchers thought the Nuremberg Code was irrelevant to the research that they wanted to do during that very period, and which they did without consent. They regarded it as irrelevant. Certainly, it's a radical view. Progress is an optional goal. It sounds so radical that it is often treated as an extreme position, acceptable perhaps to be held by a philosopher, especially one who had witnessed the 20th century's barbarities so closely and thought about them so deeply, but not relevant as a guide to policy and practice. But that view is clearly wrong. Jonas's proposition is actually the prevailing view. And if a Rip Van Winkle investigator from pre-1966 era of medical triumphalism and professional autonomy were plopped down in the world of biomedical research today, he or she would certainly feel the same way. Do we not slow down progress, or at least individual steps that individual researchers believe would move us in that direction? Every time we insist that an investigator must formulate his or her research plans in a way that allow a range of issues which they regard as salient and perhaps insist on various changes to those plans, to say nothing of the delays and notifications caused by DSMBs, IBCs, IACUCs, escrow committees, and so forth. All of these make the drive to achieve biomedical progress in the prevention, diagnosis, and treatment of disease secondary to other goals, other considerations of what makes life good and society livable and just. Jonas is simply spelling out in a dramatic fashion the conceptual basis for the apparatus of ethical review that grew up in the past 50 years. If his statement sounds more radical, it is either because we have understood the review mechanisms in a more modest and practical fashion, as concrete responses to particular problems, Tuskegee, Willowbrook, tea room trade, Milgram, the Jewish Chronic Disease Hospital, ionizing radiation, embryonic stem cells, and so forth, or because we recognize and reject the implications of actually putting any field of science, even one that takes human beings as its material for research, into a lower place in the value hierarchy. So now let me combine this with a look at the other part of my topic, young patients. Research with children, I think, crystallizes the ethical dilemmas that I've just developed around the issues of research. How should decisions be made when potential research subjects are unable to consent for themselves? Is special research needed to create and refine appropriate treatment because this group constitute a distinct population, whether metabolically or developmentally or in other ways? And then the question, is their identity as a group sufficient to justify research on members to benefit the future whole? That's the when question again. Now, up until the 1970s, I think it was generally accepted in medicine, law, and ethics that children could not be research subjects except for their own benefit. And this was the view that we heard about briefly yesterday from Lainey Ross, articulated by Paul Ramsey in his famous book, The Patient as Person. The contrary view, however, began to emerge in the 1970s, and in particular, another religious figure, Richard McCormick of Georgetown, held that it was acceptable when necessary to advance the interests of children to conduct research, and that parental consent provides the necessary protection. And the rationale is that children who are consented to participate in research are in effect being guided in their moral development because they're being shown that it is right to act to help others. In 1978, the National Commission, in its report on, in its Belmont report, said effective ways of treating childhood diseases and fostering healthy development are benefits that serve to justify research involving children, even when individual research subjects are not beneficiaries. They came down clearly on the side that this was a group, you should think of it as a group, and individual members of the group can be made instruments for benefiting the group. They took the McCormick view. And they said, research also makes it possible to avoid the harm that may result from the applications of previously accepted routine practice that on closer investigation turn out to be dangerous. Well, that is precisely the justification now given for research on practice generally, for the learning health systems. And in its report on research involving children, the National Commission then spelled out how this should happen. First, conduct research on animals, then on adults, and then on older children. Equitable selection of subjects and minimization of risks. When those are fulfilled, that's acceptable. Then, in subpart D, HHS spelled out rules for research on children. Where there is minimal risk, research may be approved. If it's more than minimal risk, but no prospect of direct benefit, you can still approve, as those of you who sit on IRBs and submit research proposals to IRBs know, if there's a minor increase over minimal risk, and if it yields vitally important knowledge. No one, I think, has ever yet defined what a minor increase is, but we all use the terms. And if there's a greater than minor increase, in other words, a greater than this thing that we don't know what it is, if it provides reasonable opportunity for generalizable knowledge and contains sound ethical principles. So I'm saying that just as we have gotten to the point of saying, do we really want these research rules? Is it time to re-examine them as to everyone? We've already seen that illustrated as to children. We have decided that the value of the research to the population justifies research without consent, even when it involves more than minimal risk. So let me end then with a set of 4 questions. The first one is, have the problems and scandals of the past created undue burdens on conducting research in the present? That is the criticism. That's the central criticism of those like Whitney who believe that we should not have IRBs on OHRP. Are the present calls to dispense with consent and review simply manifestations of a compulsive drive for progress, which is dangerous rather than sacred? Should our answers to the question, when may a society expose some of its members to harm in order to seek benefit for others and for society as a whole, which is the hard part of the question, be different today than at Nuremberg or than in the Belmont Report? And finally, when consent is impossible, as for children, do we need a new explicit discussion of Hans Jonas's challenge to us that research is an optional goal, not obligatory? And so as we look at that chart and look at the low points of the pre-1900 experiments and the World War II experiments, and the higher points when the law began to come in and when Nuremberg existed, although there was some retreat from it, and then Beecher and the Belmont Report, down to what has seemed to me to be the compromises brought about by the Common Rule and its application. We have to ask then, where are we headed? Are we headed to a new social contract where we will explicitly identify that certain kinds of research can be conducted when they are approved in a certain fashion without consent, and we are all in agreement that that's a social contract that makes sense? It has enough payoff and involves, with appropriate review, enough risk that we put consent aside and have a social mechanism? We do that in public health. This is not a radical suggestion. Or do we go back to the world of individual physician or institutional discretion, relying on the good intentions of those who conduct the research? That, to me, is the question as we enter the period of the 7th paradigm. Thank you all very much.
Thank you, Alex. Always a good way to end a conference with 4 easily answerable questions. So just a reminder, we do really appreciate your evaluations. We pore over those, take them very seriously, use them to improve the conference from year to year. So please, as you leave, turn in your evaluations. If you've remember or have not remembered to sign in for CME credit, please do that. And for those of you interested in either sharing the conference or watching it again, the videotapes of the presentations will be available on our website, and probably by late August posted on that website. So if you're interested in, or you missed one or two, feel free to go back to the website and see those. And then finally, please join us again next year. We will be back here at Bell Harbor July 24 and 25. And our topic will be the ethical issues that arise in the patient-provider and parent relationship. So thanks again, everybody, for coming. We really appreciate you joining us, and enjoy the afternoon.